Co-chairperson of the webinar, Dr. A.H. Jana sir, Principal, SMC College of Dairy Science, Kamden University, Anand. Coordinator of this webinar, Dr. Subrata Hathi, Assistant Professor at Dairy Microbiology Department, SMC College of Dairy Science, KU, Anand. Co-coordinator of this webinar, Dr. Srija, Assistant Professor and Head of Dairy Microbiology Department, SMC College of Dairy Science, KU, Anand. Dr. J.B. Prajapati sir, retired Principal and Dean of our college, Chairman, Indian Dairy Association, West Zone, and Coordinator of Swedish South Asian Network on Fermented Foods. Distinguished members from BIREC team, invited eminent speakers, learned delegates from industry as well as academia, faculty of various dairy and food science colleges, dear students, and all the participants who joined us virtually. I, Dr. Mittal Kathiria, coordinator of this two-day webinar and assistant professor at SMC College of Dairy Science, feel privileged and honored to welcome you all on behalf of Dairy Microbiology Department, SMC College of Dairy Science, Kamden University, to the first day of two-day webinar on biomanufacturing of probiotics and fermented foods, a step towards health status and social well-being. We feel very privileged to have with us chairperson of this webinar, Dr. N.H. Kelawala sir, Honorable Vice Chancellor, Kamden University, Gandhinagar. Sir is very able administrator and excellent researcher. Sir is basically veterinarian and he received numbers of awards and research grants during his professional career. Sir also published several research papers in reputed journals of national and international repute. So I now welcome Kelawala sir, Honorable Vice Chancellor of Kamden University and chairperson of this webinar for the welcome address.
Good morning to one and all. Am I audible?
Yes, sir.
Am I audible?
Yes, sir.
Yes,
you are
sir.
audible.
Good morning to one and all.
Hear you.
In this 2 days webinar, co-chairperson Dr. D.B. Patil, Director of Research of Kamden University, Dr. Atanu Jana, Principal, SMC College of Dairy Science, Coordinator Dr. Subrata Hathi, Co-coordinator Dr. Srija, Dr. Mittal, all the participants. I have been given that almost more than 800 participants have registered themselves. Faculties from BIRAC, New Delhi. I have gone through all the learned lecturers. There are 6 lectures in this 2 days webinar. So I am in pleasure to be here for the opening remarks for today's webinar on biomanufacturing of probiotics and fermented foods, a step towards health status and social well-being, organized by Dairy Microbiology Department of SMC College of Dairy Science of Kamden University and BIRAC, New Delhi. With great honor and pleasure, I welcome you all to the 2 days webinar. Dairy Microbiology Department of SMC College of Dairy Science has been working on fermented milks and probiotics last 3 decades. They have developed technologies for preparations of dairy, lassi, Greek yogurt, cottage cheese, etc., and also maintain the culture collection bank for providing the dairy starter cultures. India has a rich tradition of fermented milk products. Dairy is a staple food in Indian diet and is consumed in various forms, including as a side dish, in lassi, a yogurt-based drink, and in traditional dishes like curry and raita. Traditional fermented foods play a significant role in various cultures around the world, offering a wide range of benefits to both individuals and communities. Fermentation is a natural process that involves the breakdown of food components by microorganisms like bacteria, yeast, and molds. This process not only transforms the flavor and texture of the food, but also enhances the nutritional value. Fermentation has long been used as a method of food preservation. The fermentation process creates an acidic or alcoholic environment that inhibits the growth of spoilage and pathogenic organisms helping to extend the shelf life of the food. This has been crucial in societies with limited access to refrigeration and other modern preservative methods. Fermentation can significantly increase the nutritional contents of the food. Microorganisms involved in the fermentation process produce enzymes that break down complex compounds, making nutrients more bioavailable. For example, fermented dairy products like yogurt and kefir contain higher level of certain vitamins, minerals, and probiotics than their non-fermented counterparts. As per one intelligent report, the fermented foods and beverage market size is expected to grow from 183 billion US dollar in the current year and is projected to register a CAGR of 6.7% over the next 5 years. Many traditional fermented foods are rich in probiotics, beneficial bacteria that can positively impact gut health. These probiotics can aid digestion, enhance nutrient absorption, and contribute to the balanced microbial environment in digestive system. Fermented foods such as kimchi, sauerkraut, miso, and yogurt are also known for their probiotic content. The use of traditional fermentation techniques can contribute to sustainable food practices. Fermentation reduces food waste by preserving excess or seasonal produce. Additionally, the process requires minimal energy input and often involves locally sourced ingredients, supporting local economics and reducing the carbon footprint associated with food production. Fermentation is a versatile process that can be adapted to local ingredients and environmental conditions. Different regions have developed their own unique fermented foods based on the availability of raw materials, climate, and cultural preferences. In conclusion, traditional fermented foods serve as more than just culinary delights. They represent a combination of science, culture, and sustainability. As interest in natural and traditional food practices grows, the appreciation for these fermented products continues to rise globally. The market for fermented milk products in India is diverse, with a range of offerings beyond traditional curd. This includes probiotic enriched yogurts, flavored yogurts, buttermilk, chas or tart, and other fermented dairy beverages. Companies often introduce innovative products to cater to changing consumer preference. In this 2 days webinar, there are 6 expert lectures being delivered by experts. I request all the delegates, as per information, almost 800 delegates registered. There should be discussion and should be two-way discussions, so that whatever points to be discussed by our delegates, they can have their own ideas and any queries from the experts. And now startups are coming up. So by this means of webinar, you can have startups. And by this 2 days webinar, I wish all the success for this webinar. Thank you very much.
Thank you so much, Kelawala sir, for your welcome address. Dr. Sujit, DB Patil sir is with us or not? Because we are not able to see him.
Let me check, ma'am, 1 second. No, I can't see him.
Yeah, I am available.
Are you there? Sir, can you?
I joined.
Yes. Okay, sir.
Okay. Sir, can you switch on your camera webcam?
Yeah, but in view of connectivity, I want to avoid that. Right.
Okay, sir.
You are not getting proper voice. I think I am not getting proper voice. It is cut down words. Is there?
Yes, sir. Okay, then we'll move ahead. We are very much grateful to have with us Dr. DB Patil sir, Director of Research and Dean PG Studies, Kamden University, Gandhinagar. Sir has over 32 years of experience in teaching, research and extension. Sir has published many research articles in reputed international and national journals. Sir is also recipient of list of awards for scientific contribution in the field of veterinary science. I request Dr. DB Patil sir, Director of Research and Dean PG Studies, Kamden University, Gandhinagar, and co-chairman of this webinar for the short message.
I am audible?
Yes, sir.
Yeah. Good morning.
Clearly.
Good morning, everybody. Honorable Vice Chancellor, Kamdenu University, Dr. N.H. Kelawala, Dr. J.B. Prajapati, former Professor and Head and Principal, SMC College of Dairy Science, Anand, who has nurtured the Department of Dairy Microbiology for so long, Dr. Atanu Jana, Principal, uh, Dr. Srija, Dr. Subrato Hati, and faculty members and participants of this webinar. At the outset, I am thankful to DBT Bairek for selecting Kamdenu University for this very important webinar topic, biomanufacturing of probiotics and fermented foods, a step towards health, status, and well-being. So, the Department of Dairy Microbiology is known throughout India and also abroad. They are working on pro, pre, and symbiotics bioactive peptides for more than 30 years. They have dairy starter cultures, bank, and also develop probiotic fermented foods. All of you know that probiotics sustain gut health. The source of probiotics in India is varied from yogurt to buttermilk to curd. Coming to the business scope for probiotics, it is very dynamic in nature in view of product availability, consumer demand, and industry innovation. However, of late, there is increasing evidence of interaction of microbiota and pathophysiological processes of disease in human beings. And this is leading to increased usage of probiotics in day-to-day life. The advances in biomanufacturing are it's evolving, like fluidized bed drying, vacuum drying, coating, use of hydrogels, micro encapsulation. Of late, they are trying to introduce probiotics in chocolate, in banana powder. The list is long and there is lot of scope. And the new thing is they want to use probiotics topically for treatment of skin disorders in human being. There are some encouraging results. What is required is more clinical evidence, more validation of the clinical findings so that the clinicians increasingly prescribe probiotics in day-to-day clinical practice. This is the need of the hour, and also there is requirement of use of local cultures in India. That is also another important aspect. I am sure the deliberations in today's webinar will give new guidelines or suggestions for improving Thank you very much. I thank the organizers for giving me this opportunity. Thank you very much.
Thank you so much, Patil sir. We are very grateful to have with us Dr. Jana sir, Principal SMC College of Dairy Science, Kamdenu University, Anand, and co-chairperson of the webinar. Sir has experience of about 38 years in teaching, research, and administration. Sir guided so many MTech and PhD students. He also published number of research papers in highly reputed national and international journals. Sir also has numbers of prestigious awards to his credit. People even recognize him as the master of cheese. So I now invite Jana sir, principal of our college and co-chairperson of the webinar, for the short message.
Thank you, Dr. Mittal. Good morning to one and all for this 2 days webinar, Biomanufacturing of Probiotics and Fermented Foods, a Step Towards Health Status and Social Well-Being. We have already heard the opening remarks by the chairperson, Dr. N.H. Kela, Gandhinagar. We also heard one of the co-chairperson, Dr. D.B. Patil sir, Director of Research and Dean PG Studies, Kamdenu University, Gandhinagar. And they spoke again on what is the significance of knowing about the upcoming research outcomes for this particular probiotics, and especially since this post-COVID era, people have come to know what immunity means to all and how we can improve the same. And immunity comes again from the gut health. So I welcome again all the coordinators, Dr. Subrata Hati, co-coordinators, Dr. Shrija, Dr. Mittal Kathiria, Dr. J.B. Prajapati, sir, former principal and dean, SMC College of Dairy Science, under whose nurturing our dairy microbiology department of SMC College of Dairy Science. Well, since I am from SMC College of Dairy Science, Anand, let me brief you, those participants who are not aware of it. We have started inception since 1961, and we have been christened as the Center of Excellence for Human Resource Development by the joint ICAR NDDB panel. Currently, we are sitting at Vidya Dairy, which is again part and parcel of our 4-degree course curriculum, where our final year students are doing their hands-on experience in a commercial ISO HACCP certified plant, backed by all NDDB, Amul Dairy, Gujarat Cooperative Milk Marketing Federation, and in which the fermented milk plant was commissioned, inaugurated by Dr. APJ Abdul Kalam himself. And we are also from Amul producing the probiotic products, fermented products. Whatever speakers will be speaking on whatever outcomes they have already worked since ages, and the gut health, the nexus between the brain and gut health, showing what is the significance on that, even on our mental health and status. And as the topic says, a step towards health status and social well-being, our dairy microbiology department has worked a lot on fermented milk products, the starter cultures, the prebiotics, probiotics, symbiotics, and even the bioactive peptides as a result of the fermentation, and the clinical trials on what health upliftment they can give if our consumers goes on taking every day little amounts, at least 200 ml every day for say years together. So with this, I wish this particular 2 days webinar a great success. All will be learning from the luminant speakers who will be speaking on this particular topic. I also congratulate Dr. Sujit Das and Dr. Shilpa Gupta, who are also there as coordinators for this. Thank you, one and all. Thank you.
Thank you so much, Jana sir. I now invite a person from BIREC team for throwing some light on role of BIREC in fostering triple P model.
I'd request Dr. Shilpi Gupta, DGM Technical from BIREC, to start with your presentation, ma'am.
Thank you, Sujit. First of all, I would like to thank the organizers for organizing this webinar on behalf of BIREC. This is a very important area, and we acknowledge its importance, and it was very evident from the way the number of registrations that were obtained. And I really hope that this 2-day webinar in the month of January and another 2-day in the month of February will help in the young minds to be more inclined toward this area and venture into entrepreneurship. Before we actually start with the technical talks, we wanted to give our audience and participants and organizers a brief idea about what BIREC is all about, and how is it that BIREC is promoting the entrepreneurship mindset within the country. Sujat, are the slides visible?
Yes, yes, ma'am. It's visible.
Okay. So it was the year 2007 when the Department of Biotechnology was working towards the National Biotechnology Development Strategy. And at that time, it was ideated that there should be an organization which could nurture industry R&D. There was a lot which was being done for the academic institutes, but nurturing the industry R&D was something which was need of the hour at that time. And under that development strategy, BIRAC, the idea that an organization such BIRAC should be there, and accordingly it was set up as a public sector undertaking in the year 2012. And it was BIRAC was set up as a Government of India Enterprise Section 8 not-for-profit company, which supports startup entrepreneurs and small and medium enterprise to develop affordable products. We not only provide funding support, but also incubation support, technical and business mentorship, and several national and global platforms for our grantees to take their research forward. All the areas of biotech, including healthcare, agriculture, industrial biotech, sanitation, are supported under BIRAC. We were developed with a vision and mission which clearly indicated that it is very important to enhance the research innovation capabilities of the Indian industry, and ultimately it should result in sustainable viable bio enterprises which could result in creation of affordable products within the country. And ever since the inception, BIRAC has been energizing the ecosystem within the country. We support projects in the area, right from ideation for the development of proof of concept, validation, and also for any product which has reached advanced stage for manufacturing and commercializing. These acronyms which you see here, Sitare, EUVA, BIG, CIPRI, etc., they are all the different names of our funding schemes, and all cater to whether it's an enterprise or it's a startup or an entrepreneur. There are different schemes which are available under which the projects can be applied and funding support can be obtained. And as I mentioned previously, getting engaged with BIRAC not only provides funding support, but also a lot of mentoring in terms of technical and business mentors, and incubation space that we have created for startups to work is also being provided. All across area of biotechnology support is provided, which include vaccines, devices, development of medical diagnostics, bioinformatics, different drugs, agriculture-related area, biosimilars, and energy and environment theme areas on which the funding support is provided. Coming across quickly to the funding opportunities that we have available with us. So as I've been mentioning, we support individuals, startups, small and medium enterprise, biotech companies, research institutes, universities, NGOs, and societies. And the funding assistance is right from ideation till scale up and pre-commercialization. So quickly coming to one of our grant which we have for young student and researchers, we call it EUVA, which is Empowering Youth for Undertaking Value-Added Innovative Translational Research. This program was initiated to promote a culture of applied and need-oriented societal or industry research among young researchers. So this is a funding support for undergraduate, postgraduate, and postdoctoral students. There are EUVA centers across the country, which we have established, and these are basically housed in a university or an institute, and they are mentored by our existing bio incubators, and the fellowship is provided to young fellows, could be either at the postgraduate level or could be at the undergraduate level, for working on translational research. So basically the aim is to initiate entrepreneurial mindset at the grassroots level, so that once these students start working on these translational they can then take forward their research in the form of an enterprise. Coming to forward to our Ignition Grant, which is one of the flagship programs of BIRAC. This program was initiated to support early stage funding for entrepreneurial individuals. Basically, till that time when the big grant was initiated, there was no program available in the country wherein if any young individual has an exciting idea and he wanted to develop an enterprise, there was no platform as to how to take it forward. So keeping in mind that the BIG grant was initiated, wherein if an individual or an entrepreneur has a very exciting idea, then they can apply to this scheme. And now it is one of the most sought after schemes at programs at BIRAC. In this, grant and aid of 50 lakhs is provided for a duration of 18 months. And the applications have to be submitted to the BIG partners who are, there are different incubation centers across the country to whom the partnership has been given. And then, within a span of 18 months, the ideation, the sparking idea which any individual or a startup has, they can be taken forward for development of proof of concept or early stage validation. Social Innovation Immersion Program is another program which we have, which is a fellowship award program, again for creating a pool of social innovators. So these social innovators, they identify the needs and gap in a societal problem, and then they try to develop an innovative product or a service through which the societal issue could be, taken care of. And this is again an 18 months fellowship cycle for entrepreneurs or individuals, which can, then be taken forward if they want to set up an enterprise, or if they want to develop their own startup, then the idea could be taken forward for developing into product. So again, we have 9 Sparsh centers across the different across the country, and they work on different theme areas. And currently, more than 70 social innovators, they're working on various thematic problems of the society and trying to find innovative solutions for it. The SBIRI and the BIPP programs are, funding schemes which are meant for once you have reached till the stage of a startup or a company. So, once a proof of concept has been developed, and again, for there's fund requirement for taking it forward along the innovation chain, like you want to validate your program, or you want to scale up your product, then you can apply to these programs. These are royalty-based grants. And again, the applications can be done by Indian company, a startup, or a limited liability partnership. These could be done either alone or if in collaboration with academic institutes as well. Both these programs have a different funding methodology, wherein SBRI, we give 50 lakhs upfront as grant in aid, and then remaining product cost is divided between BIRAC and company. For VIPP program, this is for high risk technologies, and validation, demonstration, and pre-commercialization technologies are taken up, and the total cost of the project is divided equally between the company and BIRAC. So now, every stage there is of a product commercialization, we realize that there is always, the companies get stuck up at some point or the other. So once you have done validation, again, funds are needed in order to for market launch or large scale commercialization. So keeping that idea in mind, we had this program which we called as Product Commercialization Program Fund. And the fund in this program is given to technologies which have reached late stage validation, and now they are looking support for a market launch or a large scale commercialization. So this is one of the, so this will help you in taking your product finally to the market. And there is a payback of 150% of the total grant that we give to BIRAC within 8 years. But like the final milestone in any product reaching to the market is, the scale up and market launch, and this schemes helps us in taking forward those products. Also till now I've been talking only about the industry support that BIRAC gives, but there is another scheme which we have, which we called as the PACE scheme, promoting academic research conversion to enterprise, in which the academic institutes are also encouraged to develop their technologies and take them forward to validation. Because, it was realized that there are a lot of academic discoveries which are lying in the academic labs, and they don't move forward because the industry doesn't want to put in the money, or, the validation cannot take place because facilities are not there. So for that, we launched this scheme wherein the academic institutes can then, collaborate with the industry partner and either take support for validation of proof of principle, or to take forward the validation of a product in association with an industry. So once an industry, does the validation by themselves, they then get an idea that the product is working, and this would help in promoting the industry academia collaboration, and also give confidence to the industry to take forward the academic research. Till now, different schemes which were there, they were all grant based, and the fund was given in the form of a grant. But slowly we realized that it is important to make the startups and companies also realize the value of their product. So we started with the equity based funding. There are 3 programs which we have in which the equity based support is given. They are called as the Seed Fund, Leap Fund, and ACE Fund, and they are different equity-based support which is given to startups and for scaling up their enterprise. So the equity helps them in helps them in taking their product forward to the venture capitalists, who then get further interested in their technology, and if the VCs are interested, then the companies could help in getting more funding support for their product. So we have also developed a lot of nurturing ecosystem within the country, wherein bio incubators have been supported. So these bio incubators, they provide incubation space, high-end equipment to the startups, and let's say if there is any individual who could not find a lab where they could work, so they could incubate in these bio incubators and use the equipment and space available over there for taking forward their research programs. So there are a total of 75 bio incubators that have been supported within the country by BIRAC support. Another program which we have is an early translational accelerator, wherein we realize that there are a lot of young academic discoveries which just remain as a publication, and whether there was any possibility of converting them into commercially viable ventures or technologies. So for that, we developed early translational accelerators. So currently there are 6 accelerators within the country who work in different theme areas, and they try to pick up the different technologies and do a sort of third-party validation on them within their premises, and then get an industry on board to take forward those technologies. So these 6 accelerators are working in 3 different thematic areas and taking forward the academic discoveries. Also, we have a number of regional centers within the country, because BIRAC is based in Delhi, but then we thought that there is a lot of capacity building, mentoring services, and engaging with the regional communities is required, for which a center which is associated in a different state would help. So we have 4 regional centers within the country, and they help to create awareness event, workshops, and entrepreneurial challenges, boot camps to promote entrepreneurship in the biotech sector. We have a Make in India, Startup India cell. There are a lot of national, international partnerships which BIRAC has. We also have a biopharma mission program running on, in which early stage of biopharma development is taking place. Also, there are a lot of niche area meetings that we keep on doing from time to time, which helps, in which we interact with a lot of academic institutes, industry, and government organizations, which help us in knowing which is the area in which further funds are required. We also have a patenting and technology transfer for harnessing innovations within the country. So we provide advisory and financial support for filing the patents, and this is specifically for our grantees. We also have established several technology transfer offices within the country, which will help to strengthen the biocluster ecosystems. And these technology transfer offices, they help in mentoring, nurturing, and technology dissemination. And they also provide assistance in transfer of a technology, and licensing support is also given. There are 7 different technology transfer offices within the country, and they cater to different states and keep on looking for technologies which could be licensed. There are a lot of hands-on trainings and webinars that we keep on doing from time to time. These were specifically are developed as a part of capacity building vertical of BIRAC, in which it was felt that such hands-on trainings and webinars will help in building up the the capabilities of research personnel working in different areas of biotechnology. Over the 12 years, this is the impact that we have. We've been able to bring 800 products to market. There are close to 1,000 IPs which have been filed through BIRAC support. There are 75 incubation centers, and we have been able to support more than 4,000 startups and other beneficiaries. So if you have an exciting idea and you want to take it forward to the market or to develop a product out of it, you're most welcome to BIRAC. Thank you very much.
Thank you so much, Dr. Shilpa ma'am.
Just to add, I'll quickly follow on with Dr. Shilpi ma'am. Actually, the role of BIRAC in fostering public-private partnership for innovative product technology development and schemes was thoroughly discussed by Dr. Shilpi Gupta, who is DGM Technical BIRAC. So also these webinars are being organized by BIRAC as a part of mentoring support for enhancing the research and innovation capabilities of Indian biotech industry, particularly startups and SMEs. And basically the entrepreneurial opportunities in these sectors are currently hampered by lack of effective value chains, which often prevents highly nutritious local traditional products from meeting consumers' needs and reaching a wide range of consumers in both informal and formal markets. And to ensure that economic and social and environmental foundations that generate food security and nutrition for future generations, which are not jeopardized, the sustainable approaches in fermented food system will serve to provide food security and nutrition in an adequate way for everyone, which the webinar aims to follow through this 2 days. Like today, we have a theme on biomanufacturing of functional fermented dairy products, which aims to foster the development of the local indigenous technologies and ideas by expanding the scope and laying the groundwork for entrepreneurship in fermented milk sector. Also, we are looking for investment opportunities for new product development within particular health benefits and scale-ups. So the overall, the webinar series aims to create awareness of the market opportunities for fermented foods and beverages in India at global context. And in addition, the series will expand the knowledge of different biomolecules that help in digestion, antimicrobial, antioxidative, and etc. in fermented milk products. So we are honored by the presence of speakers from academia industry today. Also, we have joined by our Honorable VC sir, who was joined earlier in the morning, the Director of Research, Kamdenu University, Principal of SMC College of Dairy Science, Head of SMC College of Dairy Science, organizers. And I would also like to thank the participants who have joined us through GoTo platform and YouTube to join us for fruitful discussions. Please, if there are any queries, please let us know. You can unmute your mic and you can ask, or you can write in the chat box as well. So just to add, there is a registration of total of 1,156 a total for this 2 days. So I'm really thankful to who have joined. And I would like to request the attendees to mail us their feedback on the webinar and any suggestions for the specific subjects they would like us to cover in upcoming series of webinars. Thank you. Over to you, Dr. Mittal.
Thank you, Dr. Sujit, for making us aware about the BIRAC, and Dr. Shilpi ma'am for explaining all the programs which are available at BIRAC. I'm sure many of us will get benefited. Moving ahead. Now I invite Dr. Srija madam, Head of our Dairy Microbiology Department, SMC College of Dairy Science, Kamdenu University, and co-coordinator of this webinar for the introductory presentation on SMC College of Dairy Science. To share now.
प्रेजेंटर सुजीत को बोले प्रेजेंटर कर दिए प्रेजेंटर में है सुजीत मैडम इज इन प्रेजेंटर
सुजीत
शो माय स्क्रीन दीजिए अभी अभी आपका ये खोल दीजिए बोल लेते हैं हमने थोड़ा
Yes, we can see. We can see the slides.
स्लाइड यही है. Slide is visible.
It is
Yes.
visible,
Slide visible.
clear.
Honorable Vice Chancellor, Kamdenu University, Kelawala sir, respected Director of Research, Kamdenu University, Patil sir, respected Dr. J.B. Prajapati sir, Principal, SMC College of Dairy Science, Dr. Atanu Jana, all the invited speakers and the BIRAC members in this 2 days webinar, and my colleagues. A very good morning to one and all present here, or those who are attending this particular webinar. It's my pride privilege to present you an overview regarding SMC College of Dairy Science. The Sheikh Mansukhlal Chakalal College of Dairy Science is a pioneer institution in dairy education in India. It has been training the technical manpower for the dairy industry since last 6 decades. It has started as a grant-in-aid institution with the financial assistance from the government of Gujarat, and it started with the Indian Dairy Diploma and BSc Dairying. Later on, the college acquired the status of Faculty of Dairy Science in 1972 with the formation of Gujarat Agricultural University. And the IDD discontinued then, and the intake capacity for the BSc DT increased. The PG program in the college started in the year 1977. Later, the nomenclature of the degree has changed from BSc Dairy Technology to BTech Dairy Technology, as suggested by the ICR NDDB joint panel. And for imparting 1 year hands-on training to the BTech dairy students, Vidya Dairy, a commercial dairy plant, has started. And the college became a part of the Anand Agriculture University in 2004. And we have started a PG program in food biotechnology and a postgraduate diploma in dairy and food quality assurance. Later on, it was discontinued. A modern classroom complex was made for the benefit of the UG students. And the admission capacity of BTech raised from 40 to 67. And a fermented milk plant as well as an ice cream plant has started at the Vidya Dairy for imparting hands-on training to the BTech dairy technology students. A new sensory lab with all the modern facilities and an engineering laboratory and workshop has started in the college building. And we became a part of a Kamdenu University in April 2021. And in 2022, a PG program, MBA in Dairy and Food Business Management, was started. The college has 6 departments and 1 Dairy Vigyan Kendra, Department of Dairy Technology, Department of Dairy Processing Operations, Department of Dairy Engineering, Department of Dairy Microbiology, Department of Dairy Chemistry, and Department of Dairy Business Management. The college has trained and provided manpower to all the states of the country as well as to some neighboring countries like Nepal and Bhutan. And I'm proud to say that our alumni are placed at various positions, not only in the dairy and the food industry, but also in the administrative sector and in the banking sector, and also in various entrepreneurship activities. The college offers BTech undergraduate program of 4 years, and in the PG programs, MTech and PhD in the subjects of dairy technology, dairy engineering, dairy microbiology, and dairy chemistry, and an MBA program in dairy business management. The initial 3 years they spend at college where they acquire the theoretical knowledge as well as the practical exposure. And in the 4th year, they spend at Vidya Dairy where they are exposed to hands-on experience to develop their business skills. Vidya Dairy is an ISO certified state-of-the-art plant with a current turnover of approximately around rupees 250 crores. The plant handles 1 lakh liters of milk per day and sells various milk and milk products under Amul brand. The plant is operated by the final year BTech students of our college, and the college and the dairy are also involved in providing training programs for the technical manpower of the dairy plants of India. I'm very proud to point out that the college has a reputation of 100% of placement, and our major recruiters are the biggest dairy plants in India. Also includes the National Dairy Development Board, Gujarat Cooperative Milk Marketing Federation Limited, and IDMC. In order to have an overall personality development of the students of SMC College of Dairy Science, the Student Representative Council of the college imparts various co-curricular activities, and the students are also engaged in various NSS activities. The faculty at SMC College of Dairy Science have handled and are handling various externally funded projects by Indian Council of Agricultural Research, ICSSR, Department of Biotechnology, Department of Science and Technology, GSBTF. The SMC College of Dairy Science has a very strong alumni network in India as well as abroad, and it conducts various activities. To name few, alumni convention and a national seminar are conducted once in 2 years, and very unique events like a national dairy products judging contest and national dairy and food quiz contest are held annually. And to make the alumni aware of the happenings in the college, we publish the DSCA Newslink every 6 months. And all the activities of the dairy science has fetched the college Best Dairy Science College Award thrice. And in the context of this particular webinar, if I talk about the contributions of SMC College of Dairy Science in the area of probiotics, fermented foods, health and social well-being, I'm very proud to say that the institute is pioneer in probiotic research in India. Especially lots of work has been carried out at the Dairy Microbiology Department of SMC College of Dairy Science. The college is also the headquarters of Swedish South Asian Network on Fermented Foods, which is a professional body, international professional body comprising of researchers as well as academicians who are having a common interest in fermented foods and who are working towards uplifting the fermented foods which are available all over the world. And the coordinator of the same is Dr. J.B. Prajapati sir, and the headquarters is at Dairy Microbiology Department. The college is also recognized by Indian Council of Agricultural Research as niche center of excellence on functional fermented dairy products with symbiotics. It is also recognized as the cooperating center in the National Network Project on veterinary type cultures for preservation of biodiversity of lactic acid bacteria from the traditional fermented foods of the western part of India. The SMC College of Dairy Science is also the center of Indian Dairy Association Gujarat chapter. Lots of work has been carried out at Dairy Microbiology Department of this reputed institute. We have isolated, characterized the probiotic strains, and we are a pioneer in probiotic research, and the strains have been studied for their functional attributes also. Further, these strains have been used to develop an array of functional fermented dairy foods, and the technology for the same has been standardized, and the potential health benefits have been validated through in vivo studies, and a few of the products have been studied for their potential health benefits through in vivo human study. Metagenomic and clinical investigation of symbiotic lassi has been carried out with the medical college, and other in vivo studies have been carried out with the pharmacy colleges. In addition to that, our probiotic strains have been explored for their potential cosmetic applications as well as for oral health benefits and potential for vaginal health applications. Lots of work on bioactive peptides are happening in the Dairy Microbiology Department of SMC College of Dairy Science, especially from the milk of indigenous breeds of milk animals. And these peptide sequences having health benefits such as ACE inhibitory activity, antioxidant activity, These are submitted to the BioPep databases for the benefit of the scientific community. Dairy Science College has always believed in taking the research to the field and linking with the potential stakeholders for the benefit of the human health and the society at large. So as part of reaching out to the stakeholders, the technologies which have been developed at SMC College of Dairy Science has been compiled in the form of a booklet, and an industry meet has been held to commercialize these products, and few of the technologies has been transferred to the industry also. In addition to that, lots of work has been carried out on adulteration detection method for milk and milk products at Dairy Chemistry Department of SMC College of Dairy Science. And I'm proud to say that which has been included recently in the FSSAI Manual of Methods of Analysis of Foods, Dairy and Dairy Products. As part of reaching out to stakeholders, the faculty are increasingly involved in providing external lectures as well as visiting the adopted villages for knowledge dissemination, as well as helping out the farmers with their problems. We also serve as the resource persons for various training programs which are being conducted by the Vidya Dairy for the benefit of the dairy industry personnel. These are few glimpses of the hands-off training to women farmers on preparation of traditional fermented dairy products, which are being carried out by our Dairy Vigyan Kendra at SMC College of Dairy Science. As part of reaching out to stakeholders to increase the awareness regarding the probiotics and the functional fermented dairy products, especially to the youth, we regularly conduct a probiotic awareness day program a yearly basis, and along with the Gut Microbiota and Probiotic Science Foundation and the Swedish South Asian Network on Fermented Foods. And we have observed that with each passing year, the number of schools which are participating in the same program has increased. We also conduct conferences, webinars, and all are organized along with the SASNet fermented foods for the benefit of the academia, industry, as well as for the students. And I'm proud to point out the fact that during the COVID-19 pandemic, the faculty was actively involved in sensitizing the society about the immunity boosting foods. And as part of that, the information has been shared on social media platforms. And in the context of this 2 days webinar, I would like to highlight that some of the present research activities of ours involved exploring the cardioprotective effect of symbiotic fermented milk, anti-obesity effect of probiotic-rich fermented milk, exploring the postbiotics of probiotics and its role in shelf life extension of dairy foods as well as in human health, and bioactive whey peptides, its production, characterization, and biofunctionalities, use of spices in the shelf life extension of fermented dairy foods, as well as exploring the prebiotics and its application in development of symbiotic dairy foods. These are some of the few current research activities we are carrying out in context to the theme of this particular webinar. And I take this opportunity to thank Kamdeni University for giving us the opportunity to organize this particular webinar. And I take this opportunity to thank BIRAC for supporting us and hoping to have a better collaboration with BIRAC. Thank you.
Thank you, ma'am. Thank you so much.
Wait for something. आएगा फिर आया?
I would request the participants who have joined us through GoTo or through YouTube, please unmute yourself and you can ask your queries to us or the speakers if you have any, or you can also write your queries in your chat box as well. Please take part in this fruitful discussion. Thank you. Over to you, sir.
Stop sharing. Thank you, Srija madam. Now let us move towards the technical part of this 2 days webinar. Today we have with us 3 highly experienced and learned speakers from academia as well as industry. They are going to speak on different topics like glycogenomic of human gut bacteria, manufacturing of fermented dairy products, a mechanized approach, and biofunctionality of fermented foods in northeastern India. And then we have an open session where all the questions asked by participants will be answered by the respective speakers. Dr. J.B. Prajapati sir will act as coordinator of this session. So I request all the participants to write your queries or questions in the chat box, and your questions will be answered during the open discussion. So the first day of webinar is going to be very informative. And I sincerely hope that everyone really finds this session useful. I'm sure the knowledge and deliberation will give us new insight and dimensions for the future prospects in dairy and food sector. So let us start with very first eminent speaker, Dr. Ravinder Pal Singh. Sir is Associate Professor at Gujarat Biotechnology University, Gandhinagar. And today he is going to talk on glycogenomic of human gut bacteria. Sir, kindly start your presentation.
So do you see my PPT?
Yes, your slides are visible, sir.
Is it in the full mode, isn't it?
Yes, yes.
Okay, good morning to all of you, including the Vice Chancellor and Director of the Kamdenu Universities. And thank you very much to Ati and the BIRAC for giving me an opportunity to speak about my work, that what we do. So yes, I work on a glycogenomics of the human gut microbes, especially with the bacteria. As we know that the bacterial cells or a human cell, they have various glycosylations. So these glycosylations interact with our various receptors exposed on microbe cells as well as the animal cells. And so the interaction is vice versa. So glycosylations of the bacteria is interacting with the receptor of the immune cells. And on the contrary, the glycosylations of the immune cells is interacting with the microbial community. That's how the interaction is taking place, and that is how the microbiomes of the human gut bacteria is developed. When we say the glycogenomics, so glycans is stand for the carbohydrate, the genomics is the genome, which genomes include of the bacteria as well as the genome of the human being, so that how the interactions between our dietary fibers or a host-derived carbohydrates are interacting one to another. That's what we are going to be talking about. So if we talk about, the functional food, right, is related to the dietary fibers. It goes into our on the dietary canal, that's through the intestine. So you can see this in the functional food, the various kind of the carbohydrates are present, and all the carbohydrates has a different structure in the form. Given that they have a different structures, the whatever the applications and the impact would be a different based on the dietary type. And if we talk about the gut, so in the gut systems, we have a various microbes, including the virus, bacteria, fungi, and others. So all the microbes, they are interacting one to another. They are secreting various type of the metabolites, and those metabolites can be sensed or digested by another type of the microbes. Then they are secreting the different type of the metabolites, and those metabolites influence the development of our nervous system or all over the body. And if we talk about the gut microbiota, yes, the gut microbiota is very complex. We have been saying that there are about more than 100 trillion bacterial or microbial cells are present in our gut, relating to about 5,000 different strain or species of the bacteria. Let's say if we talk about the bacteria in terms of the phylums, this species belongs to the Proteobacteria, Tanidiides, Bacteroidetes, Actinobacteria, and Firmicutes. And majorly the Bacteroidetes and Actinobacteria, they are very potent to digest our dietary fibers, the complex carbohydrate which we are taking through our diet. And those, since we are taking a lot of the diverse dietary fibers, dietary food habits, if we say, let's say, talk about the India, we have a huge diversifications of a diet, right? So how the diversifications of the diet is influence the genomic integrity of the bacteria. So this is one of the studies where we have done, like in a standard diet. If someone is taking a standard diet since a long time, the genetic diversity would be lower. If the diet is based on the fibrous diet, of course, there will be a more amount of the starch and fatty acids. What are the starch and fatty acids? Starch and fatty acids like acetate, propionate, butyrate. So it has been shown that this SCFA, they have a very positively impact on our health. They positively modulate our immune system. They are promoting the beneficial microbial community in the gut, and they are improving the health of the human being. In the Western diet, if the diet will switch to the fast food, where the simple sugar would be higher, and the simple sugar would be higher, generally these are associated with the various diseases. Normally, the diabetes and obesity is generally increasing, and where this beneficial organic acid, right, the acetate, butyrate, propionate is generally low in amount. But if someone is switching the diet, right, so microbial diversities in terms of the genetic integrity of the microbial diversity would be higher. It means where the bacteria might be taking the gene from one strain to another strain, because through the horizontal gene transfer. So horizontal gene transfer is every second is happening in our gut, where the one bacterial species or a strain will take the particular gene from other strain and will integrate into their genome, so that they can produce some sort of the enzymes or a receptor to interact with the dietary content the individual is taking off. That's how the diversity of microbial community is increasing in terms of genetic. So as I said, the microbial communities, particularly the bacteria, they are producing the various metabolites, including the CFA, serotonin, and neurotransmitters. And generally the neurotransmitter, for example, gamma and serotonins, and with the tryptophans, they are generally modulate our brain. So how it's happening through the gut to the brain, that is the gut-brain axis. These metabolites, like if we have a fear or anxiety, the GABA aminobutyric acids, so they can try to be suppress your anxiety or a fear, or if you will take a good food, that will give you some sort of a happiness that will happen through the serotonin. That's how the interactions through your gut to the brain is happening. Like if a gut is not good, if someone has a diarrhea, the mood will not be good. That's how, again, it says that this is the various metabolites which determine the status of the individuals between the happy or agriness or in the disease conditions. So in a nutshell, it says that if someone is taking a dietary fibers, yes, the fermentations. So human being have very fantastic the ferment system, right, in our gut, where the fermentations will happen. Through the fermentations, they produce the butyrate, acetate, and the propionate, which can be absorbed through a particular type of a receptor or by the diffuse diffusions to the cell or in the blood system. Through the blood system, like for example in acetate, through the blood system is reached to the brain. So where this is a primary energy source for the brain. The propionate in generally the primary energy source for the liver and the butyrate for our colon cells. In case this metabolite or other metabolites go in a low number, so various diseases can occur, like a diabetes, colon cancer. So colon cancer can also happen where the some pathogenic population of the some pathogenic bacteria can go up. They produce the toxins and create the cancer conditions. And irritable bowel syndromes or inflammations, bowel diseases can happen if these organic acids can go in a low amount. The cardiac conditions will not be a good. Anxiety and depressions can happen. So how we can reverse these conditions, right? Yes, we can reverse these conditions. We can treat some of the patient by using our good diet or a modulations through the some of the beneficial bacteria. So how we can do that? How we can particular dietary fibers we can take, or how particular strain of the bacteria we can take in order to modulate through our dietary system or through any of the microbial community, either in the individual condition or in a consortium. We should have a very clear-cut understanding of the particular dietary fibers or a particular carbohydrate or particular bacterial strain, whether the bacterial strain should not cause any disease, whether the bacterial strain can producing some sort of the different metabolites can positively modulate the status of any of the disease. So then in order to do that, first we need to understand what metabolite they are producing. Then the second, how that particular strain is digesting that particular kind of a carbohydrate, right? If we want to modulate through a bacteria, we should understand that once we will feed that particular probiotic to individuals, how we can promote the growth of that probiotics. In order to promote the growth of that probiotics, we should to understand what carbohydrates that particular particular probiotics can digest. What could be the mechanism of that? So in order to do that, my group and we do work on deriving the mechanism of the bacterial species to see that how they are utilizing the dietary fibers, and then we also do the metabolomics to understand that what different sort of the metabolite they do produce in order to mitigate the occurrence of various metabolic diseases. Right? So if we says that, okay, how these microbes we have got, do we got on the human in mother when we were very tiny, or when we were in a mother womb, during that time we got, or we got after the birth. So of course there is not clear-cut understanding when we have got the microbes, but there are some studies suggest that when we were born, our microbiome was predominant with the actinobacteria, bacteroidetes, protobacteria, and firmicutes. And as soon as we start to take the mother milk, the number of the microbial community which is going up is the bifidobacteria, right? There are various species of the bifidobacteria present in a infant. So where the studies, one of the studies has been done over the 10 years, they found that the species of the bifidobacteria are like a longum, brevi, bifidum, aldoscientis, dentis, and animalis were higher in abundance in infant microbiome, and nearly about 80% space, 80% of the space of the baby gut is occupied by the bifidobacteria. But as soon as we will introduce the solid food, this communities goes down and other microbial communities go up. I will get back to that. So if you talk about the mother milk, so mother milk has, what are the different constituents of the mother milk? They are content. This is the mother HDR. The mother milk is content of the lactose, lipid, and HMOs. And you might be aware that all the baby or infants are not fortunate to get a mother milk. There are a few percentage of the babies, they are absent to get a mother milk. Then what we are doing is we go to the market, or mother go to the market and buy infant formulas, and those infant formulas do not have the human milk oligosaccharide, which are the constant of 5 to 15 gram per liter in a mother milk. Since those infant formulas do not have this HMOs into it, so infant formula fitted infants generally goes under the diarrhea, or the immune, or the proper development is not happening. So my group is working on the synthesis of the HMOs, so the new variant of infant formula can be developed. So in terms of the HMOs, so it's a various complex and various number of HMOs are present in a human milk. Out of that, about the 200 different structural form of the human milk oligosaccharides, which are the HMOs, are characterized, and they have in terms of the physicochemical properties, they are acetylated, fucosylated, and the sialylated. So among them, the 5 are very highly abundant. Like this says, LNTs, 2 fucosylolactose, 3 fucosylolactose, 3 sialolactose, or 6 sialolactose. So those highly abundant HMOs we are trying to synthesize in our lab. So, we generally go through the enzymatic process to synthesize them in our lab. We do use the cell system as well as the cell-free system to synthesize them through the enzymatic process. So let's say if the baby is unable to get the HMOs through our mother milk, what will happen? There will be a very low number of the bifidobacterium. So why the bifidobacterium is very important for the baby is because there are several studies which are suggesting that the bifidobacterium, they are producing very crucial metabolite, of course, including the SCFAs and other two, which positively modulate immune system, and they are helping overall development of the baby. In case where the baby is not able to get the HMOs, the various diseases can happen. Even in the later stage, the baby will succumb either to the death through the metabolic diseases. Generally, the obesity and the diabetes can go very serious. So you can see in the right side that if some baby is getting the HMOs, there is a huge number of the bifidobacterium would be there, and so that the growth of the baby will in a positive motion. Not only the bifidobacteriums, there are other bacteria known as the Rosiburia. These are also be abundant in generalies. So rhizobaria is also have a potential to digest the dietary fibers which are coming from our diet. So the rhizobaria is a connecting link when the microbial community is shifting from the initial phase of the gut to the mature phase of the gut microbiota. So Rosibaria is considering as a connecting link of the microbial switching during the different age of the baby. So if we say now we talk about how the different homeostasis of the different species of the bifidobacteria or rosiobacteria is happening. So the molecular mechanism by which the different species of the bifido and rosibaria can utilize the HMOs are known, which are suggesting that the different strain or a species of the bifido or a rosia have a different mechanism to utilize those complex carbohydrates present in a human milk. Some of the species of the bacteria, they can take this complex structure inside the cells and then convert into the monosaccharides. While some species, first they will convert into some sort of the simplest oligosaccharides, then they will take into the cytoplasm. That is how their homeostasis is getting maintained during the early stage. So we say in the suckling to the weaning period, right? So weaning period when we first introduce the solid food to our baby. In that condition, the microbial communities, even in the bacterial community, is switching from the bifidobacterium to other communities, where the total number of the bifidobacterial cells will drastically go down. It will be taken up by some other communities. So what are the other communities? Yes, we also work on that. So If we talk about our dietary food, here in dietary foods, there are various different types of the complex carbohydrates. It can have a like a xylin, arabinoxylans, the pectins, beta-glucans, and cellulose, starch, and those kind of the carbohydrate present in normal in our diet. Among those diet, my group is focused on the beta-glucans and the pectins. So the beta-glucans is already been known that they are immune modulators and they are positively or improving the cardiovascular health. So we work is here that how the bacterial communities. So in terms of the bacteria, we majorly focus on Bacteroidetes and Blotia species. So why we focus on the Bacteroidetes and Blotia, I will show you that. We work drastically on the beta-glucans and the pectin. So today I will just show you that what work we have done with the glucans given the time limit. I will not talk about the pectins today. So as I said, the beta-glucans. So there are various sources of the beta-glucans which we can obtain. We can obtain from the curdlan. So curdlan is a polysaccharide extracted from the bacteria. It's like extracellular polymeric substances of the bacteria. So lichen can be extracted from the lichen, and it can also be extracted from the marine source like a Laminaria desiccatus, the barleys, oat, bread, and yeast. These are major source for the beta-glucans. And all the beta-glucans extracted from the different source, they are different in structural property. Right. So given that they have a different structural properties, the microbes which can digest this beta-glucans, they have a different mechanism to utilize this particular type of the beta-glucan. So Bacteroidetes, so in case of the Bacteroidetes, are nearly 25 different species are known. Out of the 25, only one species is pathogenic, which is a Bacteroidetes fragilis. So Bacteroidetes fragilis is producing some sort of a toxins, and this can create the colon cancer. They also produce neuraminidase enzyme. So neuraminidase enzymes, you might be aware that the human cells, they have a N-glycosylations, right? So N-glycosylations, if some bacteria producing the neuraminidase, they can cut this silations from the glycocalyx moiety from the cells. Cell will not able to sense the dietary fibers or some of the probiotics bacteria where they have a particular receptor. So those probiotic bacteria has a receptor contradictory to the glycocalyx present on the human cells. They will not able to sense in that way, and they will not able to give the positive signaling to the cells, right? In that case, the cells can go in a disease condition. So out of the 25, only one species of Bacteroidetes is pathogenic, and others bacteria are common cells. And those common cells bacteria are generally responsible for digestions of a complex carbohydrate present in our diet. For examples, the Bacteroidetes B. that and the B. ovatus. So plenty of studies has been done with these two strains, right? And those published papers in like a Science, Nature Communications, Nature Microbiology, there are plenty of papers related to this. Those all the studies suggesting that this strains or Bacteroidetes nearly devoted 20% of genes of its genomes for utilizations of dietary fibers, and they are producing 1,000 types of enzymes to chop out that. So yes, we work with the Bacteroidetes, but we work with the other species of the Bacteroidetes. So we wanted to see that how these Bacteroidetes can utilize the beta-glucans. So we started with like a laminarin and the barley and the oat. So we screen out, we took some Bacteroidetes species and we screen out, and from that we found that the two species of Bacteroidetes uniformis will drastically grow on the laminarin. As I said, the laminarin is generally obtained from the marine source. So the bacteria was growing onto that. then we wanted to know that why this particular strains were growing on laminarin, but why not the other bacterial species were growing on the laminarin. We have done the genome sequencing of this strain, and we found that this particular strain, which was drastically growing on the laminarin, has the 3 genes, or because those bacterial species reside in our gut, if they are digesting the polysaccharide, they are not just a one gene, but they have a clump of a different gene, right? And present one to another in a together form, which is called as a polysaccharide utilization locus. So we identified the 3 polysaccharide utilization locus in particular strain, which can digest the porphyrin and agarose. The one locus, the another one is laminarin, and third one is a pustulan. So pustulans is a kind of another is a beta glucans, which can extracted from the yeast. So then we dig out that why this strain was growing on that laminarin. So this is one of the locus, right? This is a second locus where which can allow this spectrum to grow on the pustulans. So what we do is we have characterized each and every genes where we have cloned each and every genes in E. coli systems. We produce the enzymes or a receptor present in this locus, and then we have fully characterized it. And after the characterized, we have derived the molecular mechanism. So one locus which I talking about that is can utilize like agarose. You know that agarose is obtained from the agar, and you might be aware about what the use of the agar and agarose in the laboratory. So what we found is that this particular strands having around 120 kp genes is perfectly 100% matching with another bacterial species called Plabaceus, and also this particular the locus is perfectly matching with a marine bacteria. So it's very relevantly suggesting that how the human genomes was upgraded by the different genes based on the dietary style, right? So dietary styles can upgrade the genomic structures of the human. So yes, by looking into this genome, we found that whether this bacteria can grow on agar and or porphyrin or not. And indeed, this bacteria was growing on agar and porphyrin. So other strains was since but does not have this locus in another strains that was not growing on agar and porphyrin. So after doing that, all the characterization of the genes present in that locus, we derive the molecular mechanism where we so since the bacteria is a gram-negative bacteria, in the gram-negative bacteria, there are the 2 membrane, right? Outer membrane and inner membrane, and between the 2 membrane space called a periplasm. And on the outer membrane, they have in that locus, having 2 enzymes, which are the endolytic enzymes, which can digest the polysaccharide from the internal linkages. So, and then they generate the oligosaccharide, and oligosaccharide come into the periplasmic form through the SUS-D and SUS-D. These are a kind of a transporters, bring those oligosaccharide into periplasma. And in the periplasma, they have again some sort of enzymes which can cleave this oligosaccharide into monomeric form, and those monomeric go into the cytoplasm and enter into the metabolic pathways, and through the metabolic pathway they produce various organic acid, including the acetate, propionate, and butyrate, that how they positively modulate the human health. And another species we work on Blotia producta. Why on the Blotia producta? So initially people were thinking that the Blotia species, they are related to the pathogenic ones, but all are not that true. Every species is different. So we were, there could be chances that other species of the Blotia may be the pathogenic ones, but Blotia producta has been characterized as a probiotic strains. This is why the probiotic strains. One of the paper published in the Nature, they say that is producing the bacteriocin, which can inhibit the growth of vancomycin resistant Enterococcus faecalis. The paper is already published in a cell host microbes and the Natures. They can also reduce the cancers, right? There's a colitis. They improve the health in the Parkinson diseases. They do not And there's other studies has also been done that they do not have any negative impact on the various model system, animal model system, which has been carried out. Therefore, they are considered as a probiotics, and all the important of the probiotics you can read about is already been published latterly, that why they are probiotics if you are interested into it. So since the people has done that, of course they are producing the peptide in terms of the bacteriocins or the metabolites which positively modulating the human health, but there is no information about that how this bacteria is growing in our gut system, whether they can have a capability to digest polysaccharide, whether they have a capability to digest the oligosaccharide or not. We have done the drastic studies with that of the beta glucans and the pectins, that how this bacteria can grow on the pectins. And again, I'm not going to show about the pectins. So, but I'm just giving one example that how we have designed one of the molecular mechanisms by using the beta glucans with the Blautia producta. This is just an overview. I will go one by one. So we use like a beta glucans from the Lycanon and the barley. So beta glucan of barley and Lycanons, they have a beta-1,3 and beta-4 linkages. And this bacterium, again, it also have a one sort of a locus. And in locus, they have a two type of enzymes, and the one of the receptor and the controller, which can control our expressions of this locus. So one of the enzymes produced on the surface which digests this polysaccharide form of barley beta glucans, oat beta glucans, and Lycanons generate some sort of the oligosaccharide. It has a receptor, ABC transporter type of the receptor, which bring those oligosaccharide into the cytoplasma, and this cytoplasma again digest by some sort of the enzymes. Then through the metabolite pathways, it generate the small chain fatty acids. So how we have done the studies, I'll go in a very detail about it. So what we done is we first figure it out that, okay, this strand is growing on the barley beta glucans and the Lycanon. Then we look into the genomes of these ones and we dig out one of the locus. So in this locus, it has two enzymes and ABC transporters, and one is a regulator. We done the proteomics to see that what other genes might be upregulated when we are growing with the barley beta glucans. So we find the more genes, the 3 more genes, which are cytoplasmic genes, and all the 3 genes are related to enzymes. We have also done the RT-PCR to see that what genes are upregulating when we are growing with the beta barley glucan. So all the genes which are of the locus were upregulated when we grow on the barley beta glucans. So first we started that the one of the enzyme which was on the cytoplasmic membrane, and other enzyme will also sit with the locus which is cytoplasmic. So we wanted to know that, we wanted to demonstrate that is that really this enzyme is present in the extracellular part, or whether this enzyme is present in the intracellular part. So we have customized polyclonal antibody with that, and then we have done the confocal studies, and we have confocal studies suggesting that the GS16 enzymes is present on a bacterial cell membrane, but there were no signals with a one of the enzyme which is galactoside hydrolase family 94. We could not get signal from this ones because the polyclonal antibodies cannot go inside the cells. So there was no signal into the confocals. So these studies were confirmed that the one enzyme is present on the cell membrane, where the other enzyme present in a cytoplasmic form. And we have tried to the crystal structure of this, and what we found then, this is one of the carbohydrate gel electrophoresis image is demonstrating that once this enzyme is digesting the polysaccharide, what sort of the oligosaccharides are generating. So this is the cytoplasmic enzymes. They have a carbohydrate binding module. There's a 4 carbohydrate module to these enzymes. So this carbohydrate modules is trapping the carbohydrates. So whenever we are having our food, and this dietary fibers is going through our alimentary canal, right? So the micro should capture those carbohydrates. So this carbohydrate binding module of this enzymes is capturing that polysaccharide from the alimentary canal and provide access to the catalytic domain of the enzyme to chop down that polysaccharide and convert it into the monosaccharide form. Sorry, the oligosaccharide form. So we are also working with this carbohydrate, that which carbohydrate is more essential, which carbohydrate binding module is important for capturing the particular type of the polysaccharide. We are still working on to it. The second one, this enzyme is generating like this 3 oligosaccharide. That what product they have generated, we have purified this product by using various analytical instruments by HPLCs and the GPC, NMR. We have functionally fully characterized what oligosaccharide they are generating. And they're generating this 3 structured and well-characterized oligosaccharide. And that's how this well-characterized oligosaccharide go into the cytoplasm. So as I said, this is a ABC transporters. And ABC transporters, they have 3 different components. But first one, which was present in the cytoplasm, in extracellular part, is called solid binding protein. Then is a membrane part and the cytoplasmic part, which has a ATP. ATP is kind of the activity which, when it goes into, by using the ATPs, they can internalize this one. So whether this the solid binding protein, it has the property to carbohydrate binding. So we wanted to know that which carbohydrates can be preferable or significantly bind with this protein or not. So we have done the biophysical analysis. Particularly we have done the isothermal titration calorimetry. This is a biophysical kind of a property where we can determine which carbohydrate is strongly binding to that particular receptor. So through that, whatever 3 carbohydrates I was showing in previous slides was strongly binding to this receptor. And other carbohydrates, we have also used other carbohydrates, for example, laminary bios, which having the beta-1,3 linkage, silo-bios, beta-1,4 linkage between the 2 glucose unit. And the Genti-bios is again is a disaccharide form of a glucose having a beta-1,6 linkages. Those cannot be recognized by the receptor. Right, so that receptor have a very unique characteristics to bind with a mixed link oligosaccharide. We have done the crystal structures again of that receptor to see that how and where it's in binding. So it has like a acetate, is a acidic form of some of the amino acids which are acidic in nature, and those oligosaccharide bind to that site of that protein. And those oligosaccharide then come into the cytoplasm, and in the cytoplasm they have one enzymes belong to the GS94. So in class galactoside hydrolase family, 94 is called as galactoside phosphorylase enzyme rather than the hydrolase enzyme. They have a different mechanism to use it. So how that classes of the 94 is utilizing. So those is GS16, yes, they generate the oligosaccharide. This like a the MS spectroscopy of those oligosaccharide, and those oligosaccharide were subjected to the cytoplasmic enzyme. And this enzyme is generating disaccharides, right? And so those disaccharides can be utilized by the another enzymes. So we have successfully done the crystal structure of this enzyme, and this enzyme not only digesting the oligosaccharide, these enzymes can also digest the laminarin, Barley beta-glucan, lysine, and this is the first enzyme in this world which galactoside phosphorylase, which we discovered, can digest the polysaccharide. No other phosphorylase so far is known to digest the polysaccharide. And those, whichever the locus I have showed in the previously.
Ravinder Pal, sir.
Yeah,
Sorry to
I'll
interrupt
just give
you.
a 5 minutes. I'm nearly completed.
Okay,
So,
okay, sir.
those locus, we wanted to see that whether the locus is abundant in other bacterial species or not. So we have screened out, this is highly abundant among the gram-positive bacteria. We wanted to know that this bacteria, the enzyme which is present in the outer cell membrane of this strand generating the oligosaccharide, whether this was generated oligosaccharide can promote the growth of other probiotic or not. We included some sort of the known probiotics, and indeed the oligosaccharide generated by this strand promote the growth of those probiotics. So whatever enzymes we have discovered so far, we use those enzymes to generate the prebiotics. By using those enzymes, we have generated the several probiotics and demonstrate that how those prebiotics can promote the growth of the probiotics. So we work like in a interdisciplinary areas from the microbiology to including bioinformatics with the analytical chemistry to carry out our work. And yes, I have a collaboration with national-wide and internationals who are supported this my works, and thank you very much again. And thank you, Prajapati sirs, to give me opportunity to present this work.
Thank you. Thank you, sir. Thank you so much for your excellent and informative talk.
Thank you.
Our next
Can I
eminent
ask-
speaker,
Can I ask a question from Dr. Singh?
ma'am, I think we'll collect all the questions and we'll discuss at the end of the technical session and in open discussion part. So it will save our time also, and all the questions will be answered at the end. If everybody is agree with this. Oh, okay, then we'll move ahead. Our next eminent speaker is Mr. Suryakant Mahapatra. Sir, are you there?
Yes, I'm here.
Sir is Deputy Manager at IDMC Limited, Anand. I invite him to share his expertise on manufacturing of fermented dairy products, a mechanized approach. Sir, you can start your presentation. And I request both the speakers to kindly complete your presentation within 30 minutes.
We also have few raised hands from the participants, so we can ask them to, for the discussion round to keep their questions so that, we can have a discussion on that, all the raised hands that I'm seeing in the box. Fine, we can go ahead with Dr. Suryakant.
No, I'm not doctor. I'm Suryakant only. I don't have any doctorate degree.
Okay, because we need you're the first
can
industry speaker of the day, so we need the inputs from you.
Can you keep me as a presenter?
Yeah, you are as a presenter. I've made you. You can share your slide, Mr. Suryakant.
Yes. Now visible? My slides are visible?
Yeah, please. You can continue. It's
Yeah.
fine.
Thank you for providing me opportunity, Kamdhanu University and Vairak as well. A warm welcome to my seminar on manufacturing of fermented dairy product, mechanized approach. First, the table of content, method, introduction, method of preparation, then commercially produced mechanized fermented products, then types of mechanized plants, flow charts, equipment used for mechanization, then typical PNIDs, layouts, field instruments, automation and instrumentation, and I'll show you a automation architecture, innovation, challenges and opportunity as well. Initially, as an introduction, milk produced products prepared by lactic acid fermentation is yogurt or a combination of lactic acid or fermentation and yeast fermentation. It is kefir. Is called fermented or cultural product. Fermentation leads to partial breakdown of milk constituents and increases the digestibility of cultured milk products. Some of popular Indian fermented milk products are dahi, lassi, then chakka, srikhand, mishti dahi, raita, many more. Traditional method, milk is heated intensively up to boiling temperature for 5 to 10 minutes. and subsequently cooled to room temperature. And previous days fermented buttermilk will be added and set undisturbed, stirred and set undisturbed, usually overnight, and the fermented culture milk is prepared. In industrial, what we do, first milk is clarified and then it is standardized. In standardization process, we are boosting the SNF in a mixer, and then will it is go to the pasteurization, followed by homogenization. After homogenization, it is again cooled to 4-degree temperature, and then a heat treatment to increase the temperature up to 42 or 45 degree for culture addition. After addition of culture, it is incubated for certain period up to acidity comes to that pH come to the culture prepared level. And once the curd is prepared, it is cooled rapidly, and packaging and storage will take place. Then commercially, there are curd, that is plain curd, sweet curd, then buttermilk, plain, spice or salted, yogurt, set yogurt, stirred yogurt, drinkable yogurt, lassi, plain lassi, sweet lassi, and salted lassi. Srikhand is also available in market. Types of mechanized fermented plants are pilot plants, that is basically for institutional dairies and for labs, and manual plant, that is small investment, small investor, that is for manual plant, not suitable for big plants. And semi-automatic plant is medium to up to 1 lakh or 2 lakh liter per day plant, and fully automatic where we are handling more than 2 lakh, 5 lakh liters of fermented plant. The initial investment is very low in case of manual plant. However, operating cost is much more. In semi-automatic, we can avoid investing some amount in automation, and rest of the thing are controlled by operator only. In automation, initial investment is automatic plant. Initial investment is much more, and however, the operating cost and saving is in other way. We are saving much in automatic plants. These are the flow charts for curd. Everybody knows how curd is made. I have shown just initial milk is standardized, and then it is pasteurized, then mixed with up to certain level of SNF, and then it is again pasteurized with the curd pasteurizer, cooled, and then again heat up to incubation temperature, then incubated with the DVS culture, and kept it for incubation for certain period up to the acidity come. And then in case of packing in cups, what we are doing, we are without incubating, we are just putting the culture and packed in the cup in flowable form, in milk form, and then it is incubated. After packing, it will go to the incubation, followed by, after reaching the acidity, will again draw the product from incubation chamber, and it will cooled immediately to retard the pH, retard the acidity action, and then transport for cold store and packaging, storage, and further dispatch. In case of sweet curd, everything is almost same. Only at the time of mixing, we are adding sugar. In case of lassi, what we are doing, we are adding sugar syrup, and then it will be smoothened, and it will be passed through. It is homogenized. Baked curd will be homogenized very well, and then it is packed in pouch, as well as in case of it is packed in cups too. In buttermilk, what we are doing, we are pasteurize the milk, and culture is added at the 42-degree temperature. Then incubation. Curd, after preparing of the curd, will start the agitation and searing and water addition. Then searing. Then if possible, we are adding some ginger, chili extract, or salt, anything we can add, and then it will be go for thermization. Thermization leads to increase the shelf life of the product. And then cooking followed by dispense, storage and dispense. Set yogurt. What we are doing? We are preparing first mix. Mix is prepared in a very without incorporating any air into the product. We are preparing the mix, and after preparation of the mix, it will be pasteurized and homogenized, and after homogenization at 42-degree temperature outlet will send the product to the tank. It is ultra clean tank. It means the tank is such that there is no outer contamination. It will be provided through a sterile vent that will keep the product the inside the vessel. It is a positive pressure. We are putting some compressed air, filtered compressed air in the incubation chamber, incubation tank. And after incubation, what we are doing? We are doing, in case of set curd, we are directly sending the product to packing at 40 to 45 degree, as well as in case of that curd, plain curd. And after packing, it will be go to the for incubation, followed by cooling, fast cooling and cold store. Stored yogurt, here everything is same, only there is a process of smoothing. After preparation of the curd, we are breaking the curd and pass through a smoothing pump and transport to another tank. And after smoothing, we'll put fruit pulp or flavor as per our recipe, and then it is packed in cup. And set Srikhand chakka. First curd is prepared and curd is drained in a muska separator or a basket centrifuge. Excess whey will be drained and it will be collected and transport, and the curd will be heated, means cooking in a multi-purpose vat where steam as well as cooling provision is there. Sugar is added as per the recipe, and the product is, after preparation, it will be cooled and then packed in cups, further cooling and storage. Equipment used for mechanization, that is deaerator, for removing excess air, entrapped air in the product. Then tanks may be insulated, uninsulated, jacketed as per the requirement. Ultra clean tank, that is for keeping the product in sterile condition. That's a sterile vent will be there. Silos, blenders. This is a shear blender, and pump, centrifugal, lobe, screw, twin screw, shear, then pasteurizer or thermizer, homogenizer, heater, cooler and chiller, manual or automatic valves. Packing equipment such as pouch packing machine, bottle packing machine, coupling machine. Some of equipment I have shown here. There is a fermenter for preparation of culture product, then mother culture as well, and pasteurizer, flow modulating valve, then butterfly valve, pumps, powder mixer, homogenizer. Deaerator. There are two types of deaerator. One is for initially after receiving of milk, the entrapped air will be removed using the deaeration vessel. In that case, the milk is passed tangentially to remove the excess, the entrapped air, and the product will be collected afterwards from the down. And in case of deaeration vessel, that is used for removing of flavor and odor. Here we are providing a condenser. The condenser will be provided with a chilled water jacket. All the off flavor and odor that will be vacuum trapped. The PNID of some of manual plant and semi-automatic and automatic plant here I am showing. This is the manual plant. Here all the operation, including the reception of milk, preparation of batch, and only pasteurizer is PID control. All other operations are using by manual valves, and there is no automation as far as automation is concerned. Up to packing, all equipment and all activities are manually handled. In case of semi-automatic, what we are doing, we are using some of automated valve. We are using some of the automated valve to control the process parameters, and some of instruments are also being used, such as preparation, batch preparation, then pasteurization, then up to incubation tank. We are using some portion of pneumatic valves, and other operation like CIP and transport product to packing, we can use manual valves. In case of automatic, here we are using mix screw valves and other single seat and butterfly valve to control the process, fully control the process as per our requirement. The plant is fully stabilized. No or minimal interruption of human is there. Everything is controlled from the control room itself. Here you can see the number of instruments and pneumatic valves are increased. Slide is layout. Typical layout of a fermented product plant. This is, we are getting standardized milk from the silos of the processing plant, and after receiving the milk, we are preparing the batch here, and after preparation the batch, we are transferring the milk to the milk pasteurizer. Here is the homogenizer. Then this is the incubation tanks, and if possible, we are directly sending the product to the cord packing area. It may be of pouch, or it may be in cup filling machines. After the packing of product, we are keeping the product in the incubation chamber. There are 2 incubation chambers we have shown. Then after incubation, it will be go to the pre-cooling room. After pre-cooling, it will be to the cold store. Further, it will be dispatched. In case of buttermilk and lassi, what we are doing, we are preparing the batch and pasteurize, and here we'll thoroughly mixing with the agitator, followed by cooling, transfer the product to the HMST. From HMST, it will be packed directly. In field instrument used for automation are flow instrument, flow switches, flow transmitters, flow indicator controllers, level instruments, level transmitters. These are basically to control and sense the flow and measure the flow, how much flow is going to inside the equipment. Then level instrument will identify how much level and how to control the level inside the vessel. Level transmitter will be there. Then pressure instruments are there, pressure transmitter, indicator and pressure switches. Temperature instrument, temperature transmitter, indicator, everything is automatic, controlled and monitored. Conductivity transmitters are there, proximity and pH transmitter, turbidity meters, weight transmitters. As per the process requirement, we are selecting the field instrument. Then automation and instrumentation. Control and automation system shall be distributed control system or high-end PLC system. The process control capability, including standard auto-tuned PID loops. The PID loops are equipment specific. Where as per the equipment need, we are providing PID loop for heating control, cooling control, then cooling tower control, cooling tower water control, everything is there. Then functional blocks or structure text or ladder programming will be there. Then software program directly, online or offline. Then capable data collection and real-time historical trend, MIS purpose. Then DCS high and high-end smart PLC are capable to handling sequential control logics through a graphical user interface, able to provide sequential diagnostic to the operator. High-end, high-speed industrial standard of fiber optics. Fiber optics are used basically where the distance is more, means liquid milk plant to fermented plant. Distance, if the distance is more than 80 meter or 90 meter, then fiber optics will be used for automation and data transfer. Industrial managed switch, diagnostic information about the healthiness of the equipment and the load of the network and the operator console. Then components of automation are power supply, CPU, main processor, input or output cards, interface modules, communication gateways, network switch, SCADA system and HMI station, networking cards. General PLC blocks are 220 volt, 50 to 60 Hz power supply cards, CPU, then communication modules, analog input and analog output, digital input and digital output. PLC blocks. And the data will be transferred to the personal computer as well through RS-232 port. PLC operation. PLC continuously monitors a system temperature, pressure, level or altitude and take necessary action in the connection with a control process. Example, already exceeds the limit value, means when we are connected to a motor or a pump, when the frequency is it is drawing more current, we can identify through automation system. It will be trigger and show some error message there to the operator directly. And if possible, we can also trip the motor at that time also. And types of input-output cards: digital input, digital output card, analog input and analog output card. Discrete or digital input signal are two-state signal, input on or off or 0 to 1. Analog inputs are 4 to 20 milliamp signals. This is a pictorial view of a SCADA system. SCADA system is a category of software application program for the process control, the gathering data and real time from the remote location in order to control the equipment. Here we can see the agitator is moving or it is idle, what is the status, what is the CIP status, everything is available on the screen itself. Typical automation architecture I will show here. Here the main control room PC are there, where engineering station, operating station, then MIS server, internet-based internal server, everything is available and connected to a Ethernet switch. It is connected to the main CPU through a CP card. And all the RIOs are communicating with each other to the CPU. Here we are, the main control room is connected to each other control room and PCs, reception and control, then lab, packing and dispatch, administration, utility, as well as the third party, means a specific equipment that is a homogenizer. Is viewable to the operator in the main control room. The basic terminology used in automation system are PLC, programmable logical circuits. Yeah.
So please wind up your presentation within 5 minutes because we are running short of time. So kindly try to complete within 5 minutes.
It will take more than not more than 5 minutes.
Okay, sir. Okay.
In RIO panels, CPU, then engineering systems, operating station, operating client server, MIS, and power supply units. Here, innovation and opportunity challenges. Automatic culture dosing. What we are doing, we are preparing after preparing the milk and keeping it up to 42 degree or 45 degree, culture is manually added. So there is chances that there will be contamination to the product. So automatic culture dosing is a technique where culture will be directly prepared and directly dosed to the inlet of outlet of pasteurizer or at the inlet of the tank, incubation tank, and followed by after dosing, it will be cleaned simultaneously so that there is no cross contamination during the process. Solids recovery. Solids recovery, that is SNF or fat will be recovered through water push, and water recovery. Water recovery, we assume that water is plentifully available, but nowadays water is there is scarcity, and if we take an example, there are lots of water requirement for a manual or automatic plant. In case of water recovery, what we are doing, we are recovering the water from CIP systems and as well as from the water push lines. The diluted phase will be collected and it will be again reused for further water treatment plant, and it will be reused again. Then energy saving. Here in case of fermented plant, lot of heating and cooling application is there. To save the energy, we are choosing the equipment that is energy efficient, that is for motors is IE3 efficiency. We are increasing the efficiency class of the motor from IE1 to IE3, energy efficient, premium efficiency. Then other thing we can do, we can save energy in terms of regeneration. Regeneration, we can regenerate from product to product or from water product to water and water to product. Shelf life. Shelf life is a challenge for every product. If we are getting a shelf life for a minimum 5 to 10 days, then we have to think again, and it is a big challenge for the industry as well. To increase the shelf life, we are providing the thermizers, and you can avoid cross contamination as well, and we can avoid incorporation air during the processing. That will increase the shelf life of the product. And another energy efficiency is if we are going for full homogenization, the homogenization will take take the energy. Most of the energy will be consumed in the homogenization, because in dairy, homogenizer is a big equipment, and the power consumption in case of homogenization is much more as compared to the other equipment. If we do partial homogenization, here what we are doing, we are partially concentrate the milk, and it will be homogenized and remixed to the skim milk, so that the sole purpose of homogenization will be solved. However, we can reduce the homogenization capacity up to 1/3. Here you can see milk processing line capacity is 30 kLph. However, we are homogenizing at a rate of 10 kLph. Here, after standardization, we are getting a mixed skim milk and cream, a standardized milk up to 18 to 20%, and it will send to the homogenizer and again mixed to the excess skim milk line, so that the purpose of homogenization is solved, and we can reduce the saving. We can increase the saving. And thank you. If anybody has any question, you can ask here.
Thank you so
you,
much,
ma'am.
sir. Thank you, sir. We'll take question at the end of technical session. Dr. J.B. Prajapati, sir, will coordinate this session also. So we'll move ahead. All the questions will be taken at the end of the session. Thank you, sir, for sharing your practical knowledge on various mechanized approaches in preparation of fermented milk products. Our next eminent speaker is Dr. B.K. Mishra, sir, Associate Professor and Head, Department of RDAP, Nehu, Tura Campus, Meghalaya. And sir is going to speak on the topic biofunctionalities of fermented foods in northeastern India. So kindly share your presentation.
That option is not coming, Dr. Sujit. Okay, it's coming. Now my slide is visible.
No, sir. No, sir.
No, sir, not yet.
Yes, sir. Sir, now we are able to see your PPT. Sir, make it full screen. Slideshow.
Respected chairman and co-chairman of this session.
Sir, it is your PPTs are visible, but make it full screen slideshow.
It is in full screen. It is in full screen.
No, sir,
No,
not
no.
yet. You have to put in the slides. No, sir. You can see your slides, but not in the presentation mode. You have to click on the slideshow button.
I can see in full mode.
No, sir, it's not yet. We can see the introductory slide only, but it's not in a slideshow mode.
Now?
No, sir.
So, can you close it and again, can you start it? I mean,
Okay.
that you can try.
Now it is in full slide mode.
No, sir, no.
No.
Here I can see on my screen it's full slide mode. Now?
No, sir.
No, sir.
आप एक बार बंद करके दोबारा कीजिए, एक बार स्टॉप शेयरिंग कीजिए, फिर चालू कीजिए ना प्रेजेंटर में जाके, एक बार स्टॉप शेयरिंग कीजिए। स्टॉप शेयरिंग ऑप्शन आ रहा है आपके पास? हाँ अभी,
हाँ सर, अभी you can share.
हाँ अभी शेयर कीजिए,
हाँ,
अभी आया।
How it is coming?
yeah, it's fine now, sir. The slide is fine. You can start. It's fine.
Now we can see, no?
Yes, sir, you can start your presentation. So we can see your full slide.
So respected chairman and co-chairman of this session, esteemed audience, ladies and gentlemen, good morning to you all. I am very much thankful to the organizers, Dr. Subrata Hati, and BIRAC representatives, Dr. Shilpi Gupta and Dr. Sujit Das, who have given me the opportunity to deliver the talk on my topic in this important webinar. I will try to overview the entire Northeastern region fermented foods biofunctionalities. So let me first thankful to Professor J.B. Prajapati sir and Dr. Subrata Hati. Under their leadership and collaboration, we have completed 3 major DBT tuning projects on fermented foods. We developed 3 products which is going to commercialize on large scale, and pilot scale production is going on. In the month of November 2023, we have also successfully organized the International Conference on Fermented Foods in collaboration with SASNET Fermented Foods Gujarat. So now I will introduce the Northeastern region. The Northeastern region of our country comprises 8 states. That is Assam, Arunachal Pradesh, Manipur, Meghalaya, Mizoram, Nagaland, and Sikkim and Tripura. And here in the entire Northeastern region, more than 70 tribes are residing. This Northeastern region, ethnically diverse population, employs unique fermentation methods for food preservation and flavor enhancement. This cultural practice creates a rich tapestry of flavors deeply ingrained in the culinary tradition of the region. Fermented foods are vital in Northeast regions, their cuisine enhancing its taste and providing nutritional benefits. These foods reflect community resourcefulness by preserving seasonal produce, ensuring year-round availability of nutrient-rich options. Fermented foods in these regions go beyond sustenance. They embody cultural identity integral to rituals, festivals, and daily life. I will show you some fermented foods samples from different states of Northeastern region. In Assam, various ethnic groups such as Ahom, Bodo, Karbi, and others engage in fermenting foods like bamboo shoots, fishes, and rice, producing delicacies like Ikwang and Pantavath, etc. These are the photographs and protocols how they are preparing these products. In Arunachal Pradesh, showcases an array of fermented foods with the Monpa, Serdukpagan, and Adi communities preserving meat, bamboo shoots, and soya beans. In Mizoram, bamboo shoot, pickle, and sarkran are very popular, highlighting the Mizo's inventive fermentation techniques. These are some photographs of these products in Mizoram. In Manipur, cuisine involves unique fermentations like Hawai jar. This is a fermented soybean paste. They prepare it and sold it locally. These are their flow diagrams, how they are preparing at their place. In Nagaland, boasts a rich variety of fermented products, which include cereal and legume-based, vegetable-based, bamboo shoot-based, meat-based, and fruit-based fermented beverage. These are the photographs, and locally they are called as akhon, colocasia species, that is called anisi, and fermented dried bamboo shoot, bastenga, fermented crabs and sesame seeds. Locally it is called tiksu. And in Sikkim, ethnic Nepali Gorkha communities have their distinct fermented items like gunruk. This is a fermented leafy greens, and sinki, fermented radish, teput, and other products also they prepare here, like fermented milk. In the locally they called somar. In Tripura, they contribute to diversity with fermented bamboo shoots and fishes. These are the locally how they are preparing. These are the flow diagrams. In Meghalaya, the ethnic fermented food samples, they prepare, basically there are two types of product they prepare. One rice-based, that is called chubichi, another soya-based fermented foods, that is called trugumbai. These traditional fermentation practices not only impart unique flavors to local cuisines, but also ensure the availability of nutrient-rich foods, showcasing the resourcefulness and cultural significance of fermented foods in the state. On these two products, we got a major product from DBT, and we are preparing these two products, that is fermented rice beverage and fermented soybean based food, that is called trugumbai. And now these two products we are preparing on commercial scales, and very soon they will go in the market on large scale. These are the starter cultures. Earlier they were preparing in very unhygienic way without mixed starter cultures. Now we are supplying them pure starter culture, and they are preparing, many of the tribes, they are engaged in this business and preparing very good product in roti. This is the protocol, how we have developed a technology to prepare in the laboratory, and this technology we are transferring to them. The biofunctional properties of this soya-based fermented food, that is called trugumbai. This laboratory-made trugumbai with lactobacillus cultures showed higher AC inhibitory activity than the product without lactobacillus culture. In terms of antioxidative properties, the highest antioxidative capacity was observed for laboratory-made trugumbai with lactobacillus cultures, which we have isolated from various fermented foods from Meghalaya. In isoflavone transformation, laboratory-made trugumbai with lactobacillus cultures showed higher bioconversion rates than the product without lactobacillus cultures. In terms of shelf life, earlier they were preparing this product and hardly the shelf life was only 1 day. Now, with this technology, they can preserve this product up to 4 days, and they can sell in the market very easily. Now, research, we have done some experimental studies on animal model, and we have checked the effect of this fermented rice beverage of Meghalaya on antibiotic-associated diarrhea on animal model. Actually, antibiotic-associated diarrhea affects 5 to 39% of the patients, posing higher risk of adults over 65 years, and with broad spectrum antibiotics use, potentially leading to extended hospital stays and increase also cost of medicines. So to mitigate antibiotic-associated diarrhea, concurrent use of probiotics is considered as a potential strategy. So from these products, we have conducted a study on animal model. We have chosen 3 lactic acid bacteria, that is Lactobacillus fermentum, Lactobacillus plantarum, and 1 species of Saccharomyces cerevisiae. And these are the microorganisms which we have isolated from different fermented foods of Meghalaya: Saccharomyces cerevisiae, Lactobacillus fermentum, Lactobacillus plantarum, Lactobacillus rhamnosus, and Lactobacillus helveticus. And this is the flow diagram of the starter culture. We have prepared the pure starter culture and we are supplying them in 10 gram, 20 gram, 100 gram, 200 gram for preparation of these products. Also, we have transferred this technology, how we have prepared in the laboratory, and now they are preparing in the same way with our products. So this is the overview of this experimental design on animal model. Antibiotic-associated diarrhea test groups. Each test group contains 6 Wistar rats. 1 ml of each fermented rice beverage sample was fed twice a day till 14th day. In group 1, we have fed the functional fermented rice beverage. In group 2, we have fed Lactobacillus plantarum and Lactobacillus fermentum in 1:1 ratio. In test group 3, we have fed Saccharomyces cerevisiae, suspended in PBS and diluted accordingly to adjust the cell counts. Next group was normal group, fed with normal diet without any other treatments. In disease control groups, we fed antibiotic mixture for inducing the diarrhea. In last group, that is standard group, we have fed loperamide drug for inducing the diarrhea. Each of the above test groups were fed with 1 ml of antibiotic mixture for 7 days, and the antibiotic associated diarrhea inhibition of all the 6 test groups was observed till 14th day. We have checked their diarrhea assessment, detection of cecal short chain fatty acids, hematological analysis, pooled fecal samples. This is the protocols. After this adaptation of 5 days period, the rats were subjected to various supplementation to check the efficacy of each product to subsidize antibiotic-associated diarrhea. Test group 1, 2, and 3. Here we have checked the fecal consistency. On the last day of the study, the test groups A1, which was fed with fermented rice beverage, and standard showed dry and normal consistency of feces with well-maintained shape and brown color with a fecal consistency score of 1, presuming to derive the antibiotic-associated diarrhea was resolved. This shows that the fermented rice beverage, that is called locally chubuchi, may be as an antidiarrheal product. Under this hematological analysis, based on the above results, our sample A1, that is fermented rice beverage, can be considered predominant over the test group in the study for resolving the deleterious effect of antibiotic-associated diarrhea. Under this histopathology analysis of colon, we have seen that test group 1, that was fed with fermented rice beverage. This group not shown any abnormal changes that indicate it could work well as antidiarrheal drug in comparison to other groups. Under histopathology analysis of liver, test group A1 did not reveal any specific lesions and is at par with the normal control test group, indicating that it could work well as antidiarrheal drug. Under cecal short chain fatty acids quantification from antibiotic-associated animal models, the cecal concentration of lactate, acetate, and propionate were significantly higher in the rats of test group A1 fed with fermented rice beverage as compared to rest groups. there were no presence of acetate in the test group, normal control, disease control, and standard. Another experiment we have done, significance of these two Lactobacillus fermentum and Saccharomyces cerevisiae isolated from fermented foods of Meghalaya on the growth performance, hematological traits, serum biochemistry of broiler chicken. We have taken 96 one-day-old broiler chickens and divided into 4 groups. In group 1, we fed control with basal diet plus commercial probiotic plus immunomodulator. In group 2, we have fed basal diet plus Lactobacillus fermentum. And in group 3, basal diet plus Saccharomyces cerevisiae. And in group 4, basal diet plus combination of T2 and T3 cultures in the ratio of 1:1. So in this study, the birds were housed at Anand Agriculture University, Gujarat, and received a standard corn soybean meal as a basal diet, meeting nutritional requirement for starter and finsearch birds. The study conducted ethnically according to Indian guidelines. In the present study, these two cultures, that is Lactobacillus fermentum and Saccharomyces cerevisiae, were considered as an alternative to growth promoter antibiotics. Broilers were grouped into 4 different groups, as we have discussed in the earlier slides. So we have checked the histological image of intestine, heart, and liver tissue of the broilers. We found histological examinations. This revealed no significant difference in the intestinal, liver, and heart tissue of the broilers fed with Lactobacillus and Saccharomyces cerevisiae compared to other control group. Microscopic examination showed well-organized epithelial linings and villi structures in all groups. In fecal counts of this lactobacilli, there were no statistically significant difference was found between the feed supplemented and the number of coliform in feces during individual week of rearing, although lower coliform was found in these groups at first week and 6th week respectively. Enterococci count was taken on KF streptococci agar plate, which was found not significant. In metagenomic study of cecal sacs of the broilers, the study found no significant differences in the relative abundance of genera or phyla among various groups. Now we will show you the biofunctionality, the role of fermented foods on human health and societal developments in entire northeastern states. Lactic acid bacteria involved in fermentation can resynthesize folate, which are otherwise destroyed during heating, showcasing potential health benefits. Native fermented foods in the northeast region of our country are believed to possess medicinal properties, offering antioxidant, antimicrobial, probiotic, essential amino acids, and low cholesterol characteristics. Functional microorganisms in fermented foods in these regions enhance the bioavailability of nutrients, improve food safety, and contribute to the degradation of toxins and anti-nutritional factors. The functional properties of microorganisms, how these fermented foods on human health and societal development in these states. So the functional properties of the microorganisms in the fermented foods include probiotic properties, antimicrobial properties, antioxidative activity, peptide production, fibrinolytic activity, polyglutamic acid production, and the degradation of anti-nutritional compounds. Certain species of microorganisms with functional properties in fermented foods are commercialized and marketed globally as functional food, health foods, therapeutic foods, etc. Fermented foods, due to the presence of functional microorganisms, offer various health benefits including nutrient synthesis, prevention from cardiovascular diseases, hypertension, cancer, gastrointestinal disorders, allergies, etc. Now we'll discuss about the role of fermented foods on societal development for the quality improvement and disease prevention. Essential knowledge for enhancing food quality. Fermented foods contribute to disease prevention. Consumer awareness grows regarding fermented food preparation. Traditional knowledge, how the impact on traditional knowledge. Native traditional knowledge in the Northeast India is very crucial. This play a significant role in fermenting food, offering industry opportunities and opportunities for food industry despite challenges. Traditional knowledge offers vast opportunities, potential for developing fermented food industries in Northeast India. Nutritional, there are several products from different tribes, these fermented foods of Northeast India. So, few popular fermented foods of Northeast India, we will show you here. Nima, this is a sticky fermented soya foods from northeastern of Nepal border and Sikkim. The main microorganisms involved in this product is Bacillus subtilis. Trungumbai, this is fermented soybean food of Khasi and Jaintia Hills tribes of Meghalaya. Major microorganisms involved in this product is Bacillus subtilis, Enterococcus, PCM, and other various yeasts. Gundruk, this is defined fermented leafy vegetable food of the Gundruk Gorkha in Sikkim. Various lactic acid bacteria and Pseudomonas pediococcus are the main microorganisms in this product. Swardan, this is fermented bamboo shoot food in Manipur, consumed by the Meitei tribes. Selroti, this is the popular ring-shaped, deep-fried fermented rice product of the Gorkha and ethnic Nepali in Sikkim. Tungtep, this is fermented fish paste of Khasi in Meghalaya. The major microorganisms involved in this fish paste is Lactobacillus, Enterococcus, Bacillus, Micrococcus, etc. Kodo Kojwar, this is the popular fermented finger millet-based beverage in Northeast India. The major microorganisms involved in this product is Mucor, Rhizopus, various species of this yeast, etc. This is Tungbai and Bekang are the traditional fermented soya foods of various states of northeastern region. In different states, the name is different. Like in Meghalaya, this is called Tungbai. Utonga, Kusup, Hentang, and Nagri, these are the traditional fermented fish paste from Manipur. This shows notably antioxidant properties with high DPPH radical scavenging ranging from 35.77 to 52.11%. Sidal, this is a salt-free fermented fish product from Northeast India. This shows significant antioxidant properties. Epong, this is a rice-based fermented beverage of Northeast India, which is rich in antimicrobial and antioxidant properties and believed to prevent kidney stone formation. Hawai jar, this is a fermented soya product of Manipur and having very high protein contents and antioxidant properties. Ekjon, this is a fermented soybean food from Nagaland, is known for its high antioxidant properties and contributing to its potential health benefits. The fermented bamboo shoots, they are very rich in phenolic compounds and tannin, having high antioxidant properties and anti-cancerous properties. Mishu, this is a bamboo shoot pickle from Darjeeling Hills and Sikkim, is rich in antioxidant, offering immense nutritional and medicinal properties. Fermented legume foods, they are integral to diets in many developing countries, including India, and also in various states of the northeastern region. In these products, anti-diabetic properties are also found. Fermented foods of northeast parts of our country, particularly those involving lactic acid bacteria, offer health benefits, including anti-diabetic properties. Bioactive peptides produced during fermentation exhibit antioxidant, antimicrobial, anti-inflammatory, and blood pressure lowering effects, and these are the contributing to enhance the nutritional value. Hawai jar, this is found in Manipur, soybean-based fermented foods. This is also having
Mishra sir,
diabetic properties.
sir.
Yes.
Mishra sir, sorry to interrupt you, sir. So please try
Yes.
to complete within 5 minutes.
Okay. And in these fermented foods, we are also claims antimicrobial properties and culture. These are the some cultural significance of fermented foods of northeastern region of our country. These are the challenges and future prospects of fermented foods of Northeast India. Preserving traditional fermentation practices in Northeast India faces several challenges in the contemporary context. The rapid pace of urbanization, changing lifestyle, and increased reliance on commercially processed foods have threat to the continuity of age-old fermentation technique. To address these challenges, efforts should be directed towards community-based initiatives, educational programs, and documentation projects that promote the value of traditional fermentation practices. This ensures their preservation as an integral part of Northeast region of the country. So I can conclude this topic on this. Fermented foods in the northeastern region of India holds profound cultural, social, and dietary significance. These traditional culinary practices are deeply woven into the fabric of daily life, playing pivotal role in religious rituals, festival, and communal gatherings. They serve as a symbol of community bonding, hospitality, and shared experiences. And research exploring microbial ecology, health benefits, and sociocultural aspects is crucial for a comprehensive understanding, ensuring the continued appreciation and preservation of Northeast India's unique fermented foods. These are the some publications we have jointly published with Anand Agriculture University and Kamden University, Gujarat, in the part of DBT tuning projects. And these are the references. So thank you all.
Thank you so much, sir. It was a really very knowledge enriching and informative presentation on fermented foods of northeastern India. Thank you very much, sir. Now it's time for the open discussion and concluding remarks. And for that, I request respected Dr. J.B. Prajapati, sir, retired principal and dean of SMC College of Dairy Science and chairman of IDA West Zone, coordinator of Swedish South Asian Network on Fermented Food, to kindly be the coordinator for this session. Sir, unmute. Sir, unmute your speaker.
Yes.
So we are not able to hear you, sir. JB, sir. Sir, you are not audible. JB, sir, you are not audible. So we are not able to hear you.
Hello.
Yes, sir. Now you.
Yes,
Now
we can hear you. You're audible.
Okay. So, thank you, Dr. Mittal. I think now we can take the questions for the speakers first. I think the first question was there from Dr. Shilpi. Dr. Shilpi Gupta ji, please pose your question now to Ravindra Pal Singh.
Yeah, thank you very much, Dr. Prajapati. So my question to Dr. Singh was, so he had shown us the data for a lot of strains that they were using for producing different compounds. So I wanted to understand, are these like normal strains, or have they been like genetically modified or recombinant strains that are being used for the study?
Okay, so whatever strains, like we are working with the bacteroides and Bluetia strains, they are not modified genetically. They are a normal human strain.
Okay, yeah, thank you. Because I was wondering, like recombinant strains would not be allowed to be used in foods, the products obtained.
Yeah. No, these are the normal flora. You're right, but actually, many people are also trying to modify certain genes and try to apply. I do not know when the time will come where we will be able to apply this modified, genetically modified culture. Yeah, I am also using the synthetic biology to modify the E. coli Nissel, which is a probiotic strain, for making the human milk oligosaccharide. But that's a completely different approach, where we will just use a carbohydrate. We will not use the bacteria. Yeah, actually, I have one question to Dr. Ravindra. Yes, sir. Actually, what is the source of those ingredients which you used? For example, beta glucan and other things. Is it a natural source, or is a like a normal barley? I am using barley, oats, whichever available in the supermarket. So what we are doing, whenever we are buying this material from the market, we generally purify in house by using a GPC or HPLCs, and then we are doing test through the NMR and the mass spectrometer just to see if we use before we use for the bacteria, whether they are highly purified and what we are think is that is or not. If we think this is a highly purified and just only the particular kind of the carbohydrate, then we are using for the acid. We do not use as a raw material. Because if you use a raw material, then whatever impact will come, we will not sure how it's coming. That is true. I request Dr. Mishra to stop screen sharing.
Okay, sir.
Dr. Singh, I also wanted to know, sorry for interrupting. So, like,
yes,
you've got these wonderful strains and they are giving very good yield and productivity for some of the compounds which are, extremely useful. So, have you, do you think you have approached a yield wherein these could be taken forward for commercial utilization?
Yield in terms of what?
Yield in terms of the amount that you're getting?
So, we have quantified the productions of the acetate, butyrate, and propionate from there. And if we talk about yield in terms of the prebiotic, which we generate in the oligosaccharide, we generate through the enzymatic process, we have also done that yield.
So then are these at a scale where they would be commercially viable? Would an industry be interested in taking forward these compounds or the process?
This is enzymes, which is, it's highly useful for the commercializations. But I haven't explored that area yet. So, which we have cloned and we have those enzymes with us. We have nearly about 40-50 enzymes which we have cloned, and those enzymes can cleave xylans, wheat, barley, beta glucans, oats and yeast beta glucans. So we have all sort of the enzymes with us. So it's our a routine work where we nearly cloning every day. So if I say in a different ways, we have cloned more than 1,000 genes so far.
Actually, Dr. Ravindra, you also told about some novel enzymes which you found out. Is a new enzyme or something like existing one with some
It's
application?
like a different variant of enzymes. Like one we found is a galactoside hydrolase belong to family 158. That enzymes can chop down the Cadlan. So Cadlan is a highly complex polysaccharide, not soluble properly because it's a bacterial polysaccharide. But we figured it out that enzymes can chop down the Cadlan. So I think this is when we are the first in the world which we shown that how these enzymes can cleave the Cadlan. So when we're working with that enzymes, the another enzymes very similar, having a 99% the homology will also be discovered from one of the Canadian group. So they also did the crystal structure and submitted in there. So the like who has discovered the enzymes goes in their name, but we were the first who discovered that enzyme. And
That's
the second one is the galactoside, that phosphorylase belong to 94 family. This is the first enzyme in the world where we demonstrate that it can cleave the polysaccharide. But so far, previously, the phosphorylases are generally known to cleave the oligosaccharides, not the polysaccharide. They do not have the pocket is very inside, like in a grooving pocket. But this in enzyme, the pocket is wide in form. So given that pocket in a wide form, it can chop down the polysaccharide. It's not the case of other galactoside hydrolases family belong to GS94, which we published in a JBC this year. This all work.
Okay. Thank you so much. Yes, congratulations. I think Mr. Suryakant is also there on the screen because we have questions for all the three speakers.
Yes.
Suryakant, if you are there, I have one question for you.
Yes, sir.
Have you taken any specific care for design tanks, pumps, action pumps? Because when you are pumping or storing fermented milk type of product, you have to see that the damage to the coagulum is minimum. So is there any change in the design of the tank, interiors?
No, there is no change in inside the vessel design.
I suggest that
you
should
are
have
saying about online dosing or offline dosing?
No. For example, maybe batch incubation tanks.
Yeah.
So that cleaning is also good and the minimum shear stress to the coagulum, especially with the stored products which are going to then pack into the cups and pouches also. For just mixing an inoculum and then packing directly in the pouch is not an issue, but for fermented milk product which you want to handle, you need to have special tanks. And also the pumps. The pumps which takes the fermented lassi or curd or buttermilk.
Yeah, pumps are different. Definitely, pumps are different. It may be lobe or twin screw pump we are selecting.
Yes, that is.
So that minimum damage to the product structure as well as texture. But inside the tank there is no development.
Yeah. Anyway, I can see that there are more than 200 people on the this Zoom call, and there are many more on YouTube. If there is any question from the persons who are on the Zoom, they can have first preference. Or otherwise, Dr. Mittal, you can read the questions from the chat box if there are there. For the.
I have a few questions noted actually. Can I share them, sir, with your permission?
Yes, Sujit, please.
First is for Dr. Ravindra Pal. This is from Dr. Bhuvaneshwari Gajendran. She's asking you, can gut microbiota derived probiotics offer a therapeutic solution for glucose dyshomeostasis induced by environmental endocrine disruptors in rural communities?
Yeah, why not? So glucose is a primary source. Generally, nearly all the pathogens bacteria use the glucose, right? And many of the beneficious bacteria, when we started to grow, so many of the probiotic strains do not prefer to use glucose. For example, I was working on the lactobacillus, Lactobacillus reuteri. They do not prefer to use glucose. They prefer to use disaccharide. Right?
All right.
So while the pathogens bacteria prefer to use glucose.
Okay. So one more we have from Isha Dave. She's asking, sir, I had a question regarding the HMOS you described. What was the reason of lower amount of Archimansia municipalia with HMOs and increased amount in its absence?
So human milk oligosaccharide is a very crucial, right? It's abundantly nearly to 5 to 10 gram per liter present in our mother milk. If they will not have, in general, what is happening, they as a primary source for the bifidobacterium. And we know that 80% space of the infant microbiota is occupied by the bifidobacterium. And some other studies also suggesting that on the cell level of the bifidobacterium, they have a glycocalyc moiety, especially the glycocalyc moiety containing the sialic acid. And the sialic acid interacting with a certain receptor of our gut epithelial cells. Thereby, they are positively modulating immune system. It means that they are activating dendritic cell and macrophage cell lines. So if there is a pathogens, they can go and eliminate that pathogens. That how interaction is taking place. So if less in HMOs, right, and the high abundance of the fucose or a sialic acid. Again, the fucose and sialic acid is also preferable source for many pathogenous bacteria, including the Klebsiellas, Pseudomonas. They prefer sialic acid. But on the other hand, bifidobacteria is also preferring the sialic acid and fucose. They can provide a competitive competitions for utilizations of particular dietary source. Is that what I'm trying to say or asking?
All right. Thank you, sir. And sir, one more that is from me and from my end only. I'll ask you one. I just wanted to understand. That is intracellular glycogen accumulation has been associated with important physiological functions in several bacterial species in gut metagenomic data to highlight the relevance of this.
Right. That bacteria is also the carbohydrate and the carbon source. That is what you are trying to asking me. Is that?
Yes, sir. Yes, sir. Correct.
So what is happening? You know that all the bacterial community, they have their own lifespan. They may survive 1 day or 2 days, and they will either die or they will multiply for their progeny. That is what is happening. And then a lot of the debris are present there, right? So if we talk about like bacteroids, they can digest this the complex storage material, whether it is present as the dyed bacterial cell membrane or they present in the bacterial cytoplasm. This eventually that will become a carbon source for another bacteria. That is how the bacteria is maintaining their homeostasis.
Right. Thank you. Thank you, sir.
Yeah.
Thank you so much. And next question is for Dr. Mishra from Sachin Rajput. He is asking that do the animal's gut also has good microbes colonization once the probiotic treatment is stopped? If not, then how long the effect of probiotics is sustained after the treatment is stopped? That's what we have a question from one of our participants. Are you there?
Yes, I can hear.
Yes.
So, can you repeat his question? What we exactly want to know?
They are asking, do the it's from Sachin Rajput. Do the animal's gut has good microbes colonization once the probiotic treatment is stopped? If not, then how long the effect of probiotic is sustained after the treatment is stopped?
Something about animals, Dr. Mishra.
Yeah, also they ask a specific animal model that you have used for the analysis. Which animal model they are asking? Dr. Priyanka Pathi.
I cannot see. Yeah.
Probiotic effects will be available in the gut of the animals.
I think some connectivity issue.
issues or the connectivity issues.
I think I can just give some comments on that if I am audible.
Yes, sir, you are audible clearly.
Actually, this the animal gut and human gut, there are not major differences with respect to microbes. Only microbes will be different in terms of the feed which the animals take. And when we are talking about larger animals, we have done some studies in poultry as well as in cattle. The lot of probiotics are available for cows and buffaloes and also, and poultry is full of probiotics. There are so many things. Only thing that the experience of the retention of such probiotics after stopping feeding, which he is asking, is in our human studies, we have seen that it is about 3 weeks. That when you are feeding for about 3 or 4 weeks, the population of probiotic flora rises, and it remains higher for about 2 to 3 weeks, and then again it comes normal. That means you need to have repeated feeding at least at the interval of up to 15 days or so. So you have to give a supplemented dose. In my observation, maybe after 15 days or so. It is what is my belief and experience.
Thank you, sir. And one more question for Dr. Mishra I just received now. This from I think Dr. Qureshi. He's asking, sir, the question is, one second, that how do you plan to commercialize starter culture for controlled fermentation and entrepreneurship promotion in Meghalaya and northeastern states?
Am I audible?
Yes, sir. Yes, sir. I'll repeat the question. How do you plan to commercialize starter culture for controlled fermentation and entrepreneurship promotion? From Dr. Qureshi, AIMS.
Actually, we have taken the samples of different starter cultures and fermented foods of the Meghalaya and other northeastern parts of the country. We have isolated the pure lactic acid bacteria and some species of yeast. After characterizing them, we are preparing the starter cultures in their media, how they were preparing in their in their tribe groups. So now we are preparing this in 20 gram, 100 gram, 200 gram, 500 gram to the major tribes, those who are involved in this business. So for the entrepreneurship development, the same thing can be trained to the persons, those who are interested to start this starter culture business. We have all these facilities in our campus, Northeastern University Tura campus. So anyone can come for 1 week and we can train them and they can start the entrepreneurship business in this area.
Okay. So do you want to ask any questions, Dr. Amir, Dr. Priyanka? Please unmute yourself if you have any questions. I've seen your raised hands in the chat box. Or Ms. Bhuvaneshwari, Dr. Joy. If you have any questions, please unmute. Also, Dr. Govind Rajan. You can unmute and ask if you have questions.
Actually, I can supplement the earlier questions posed to Dr. Mishra, because I was also going to ask same thing.
Please,
actually, we should have a facility in the northeastern region, especially maybe at your institutions, for manufacturing of starter cultures of bacteria, yeast, and molds. And those can be distributed not only to the entrepreneurs but also household. So probably my suggestion is to the DBT also and BIRAC, Dr. Sujit and Dr. Shilpi Gupta, that if you can finance some such center, we can have a good probably pharma-grade biomanufacturing facility where they can have hygienically packed cultures for yeast and bacteria that can be given to these. Probably they will become self-sufficient within 5 years. They can have a proper cell. For entire northeastern region, that will be a center. And then if it is required, it can be exported to other states and also to the other countries. So only thing is that it is hygienically cultivated in pure form. Then it is processed and packed, so that, because as it is also, I have seen that those cultures which are being sold in the market, they have shelf life 1 week or 2 weeks or so. So if they do, hygienical processing in a good manufacturing facilities with GMP and whatever GCP and everything is there, I think that will be a sustainable center in the northeastern region for distribution of the cultures for permanent foods of northeastern region especially. So probably Dr. Mishra can make a proposal. I can help him to finalize it, and then probably Deputy Bairak or any other scheme, because Dr. Shilpi has presented many projects. It will definitely fit in one of the projects or financing opportunities.
Thank you, sir. This is a very good idea, and we can prepare such type of proposals, and very soon we can submit it. In that way, the entire northeastern region, because they had yes. Thank you, sir. This is a very good proposal. So very soon we can prepare this proposal and submit to the VIRAC, and we'll take your help. In that way, Northeast region will be a big boost for this starter culture center. Thank you, sir.
Thank you. Dr. Shilpi Gupta, do you have any suggestion, or maybe Sujit, what also want to ask?
Sir, I have a question.
Maybe, like offline, we can discuss more about it as to how to go about it.
Okay. Yes.
One question I have.
Yeah.
Sir, this is to Dr. Ravindra. Sir, you have specialized focused on this most of glucose and polysaccharides for the probiotic growth. Is there any specific nitrogen sources that you would like to significantly highlight, which will be very useful for probiotic growth?
Can you repeat it, please?
Sir, actually, you're focusing more on the carbohydrate as a carbon source for the production of probiotics.
Yeah.
Are there any significant nitrogen sources which will be useful for the growth of probiotics?
I have not explored with the nitrogen sources.
Okay.
Yeah.
No, as in, are there any significant sources which will be useful, or anything critical which you
Maybe the play with the protein and to see that how the nitrogens can play.
Okay.
You can try to fit with a different sort of the protein, or percentage of the protein, and that someone's can
I
be.
think protein has to be there, but that is,
Even some amino acids can be used.
So
The varying concentration of the amino acids.
I think it is 1 o'clock, and Dr. Sijit, just questions. I can take last 5 minutes to conclude.
Sure, sir. And
Dr.
last
Mittal, I have one question for you. If I'm audible?
Yeah.
Yes.
Yeah, so actually, I just want to understand a very basic concept actually. So, lots of bacteria used to called as a probiotic bacteria, or there are lots of active ingredients which is being used with the probiotic bacteria. If we can use the probiotic bacteria in any form of drink, it will become a probiotic drink, or we have to do fermentation, then only it will become a probiotic drink. So it's like in that case, if we just have to add the bacteria and it will become a probiotic drink, then we can add to any kind of drink and it will become a probiotic drink, right? With the active ingredient. So I just want you to enlighten that thing for me.
I think I can make it clear. There is a clear-cut definition of probiotic. It's a probiotic organism. It can be bacteria, or it can be yeast, or anything. Only thing, it should have at least one clinically proven health benefit. That is number one. And then when we add such bacteria to any product, whether we ferment it or not, or we carry that bacteria through any food material or maybe pharma material, which reaches to your intestine in live form, it should function there, and it should exhibit the same clinical benefit which you have claimed for that. So it is not that whether you ferment it or not ferment it. Simply you have to carry it in live condition in sufficient numbers. So please read the FOWH definition, which is adopted by Indian Distinguished. And now in SSI also we have put that at least the product or whatever you are supplying, it should have at least 10 to 8 viable cells per serving size of the product.
Perfect, sir.
Is it okay?
Okay, sir. Thank you so much. Yes, sir. I understood actually. Thank you so much.
One more last question from Dr. Priyanka. I think she has been asking for a long time. Can you please unmute and please ask your question?
Yeah,
We'll
hi.
take the final question from you then.
Yeah.
Hello. It
hello,
was a
sir.
very informative session. Actually, I have two-part question. First is, what are the some of the major regulatory compliances specifically when developing probiotic, postbiotic, or any kind of functional food products? And second, since a lot of strata culture, actually all of them have cross-feeding mechanism between different group of bacteria or different strain of bacteria. So when we are developing a starter culture or when we are developing a product from a starter culture, are we translating the cross-feeding mechanism between those microbes to the product, or we are just waiting for the end product and then translating into the product?
This question is for me. As far as regulatory things are concerned, the first part of your questions, we don't have any regulatory measures for probiotic and probiotic foods, except that FSSI has given now some standards. That is what I told earlier, 10 to 8 cells per serving size. However, if you want to sell that probiotic as a pharma product, then this FDA regulations apply. That depends upon what kind of claim you want to make, and for that, what is the requirement of as active ingredient also. However, the second part of the question is, for other things, we have a guidelines. It's not mandatory for other things also. But when you are selling something as a postbiotic or after the cells are dead and as an ingredient also, for example, maybe as a food supplement or as an ingredient or so, maybe bioactive peptides or in from other proteins or other bacteria or so, that can be taken as a food ingredient or active ingredient, and for that you need to have a license for manufacturing it in a pharma grade facilities. However, when you want the doctor to prescribe it as a food drug, definitely FDA. So only we have this FSSI, the general regulatory practice standards, as well as the FDA. And probably both are thinking to have much stricter regulations in future.
Yeah, my point of that my question is because a lot of nutrient supplement, for example, the well marketed one is Good Buck. I'm sure everybody must have seen. So even though they are marketing something related to probiotics, a lot of claims they are making on marketing campaign is not directly proportional to the actual product they are selling. So if there are no guidelines or no mandatory regulatory compliances to follow through, so how we are going to bridge that misconnection between marketing and the product itself? That's what my query is.
There is no control over that. Only thing you have to just depend on this their reputation and then effectiveness. Otherwise, there is no control. No control over that.
Okay. Thank you.
I think we should close now, Sujit.
Yes, sir. Others, yes, sir. Others can, mail us your queries. We'll be happy to answer you, or you can come back tomorrow at 10 a.m. We'll be starting back with our other theme. So please join us back. Whoever is listening to me in GoTo and YouTube, please join us back from 10 a.m. tomorrow. And I'd like to thank Dr. Prajapati, sir. We are truly honored by your presence today. Of course, you need no introduction. Everybody knows you actually. So thanks, sir, for joining.
So just to sum up, actually we had a very good beginning today. Those who are joined late, we had opening remarks from the Vice Chancellor of Kamden University, Dr. Kelawala. He has also briefed about the significance of fermented foods in our country. It was followed by Dr. D.B. Patil Director of Research addressing, and then he has appreciated about the work which we have done, and a good presentation from Dr. Shrija and Dr. Jana about the work done at SMC College of Dairy Science. Actually, we started work in 1980 on probiotics with our leadership of Dr. G.M. Dave, who's retired and former principal, on beneficial lactic acid bacteria. And the concept of probiotics was probably a lot of spread in that. And probably Dr. Shrija forgot to mention that we have about 14 cultures deposited as Indian patent deposits at IMTech Chandigarh. And then Dr. Shilpi Gupta presented a very good information about what DBT BIRAC is doing, helping to the students. Probably I was seeing that one time about 1100 participants were there on YouTube, and probably a lot of good information has gone that what DBT BIRAC can do for the students and faculty and academics as an institution as well as individuals too. And they have many schemes befitting to the needs of different categories. And then we had a good 3 eminent speakers, probably most befitting to the theme of the seminar. Dr. Ravindrapal Singh from Gujarat Biotechnology University, Suryakant Mahapatra from industry, IDMC, that what equipment are being manufactured, and Dr. Mishra is an expert in the fermented foods of northeastern region of South. And we had a very good discussion. So probably everything was good. I thank DBT BIRAC and Dr. Shilpi Gupta, Dr. Sujit Das, and the organizers of this seminar for allowing me to speak few words and lead the discussion. Probably we'll be joining tomorrow once again. So thank you so much, and thank you all the participants, and thank you organizers. Dr. Sujit, now
Thank you,
final.
sir. Thank you, sir. Thank you so much.
Thank you.
Thank you.
Thank you, sir. Thank you for
Thank you, sir.
attracting.
See you tomorrow at
Thank
10
you.
o'clock.
Sure. Thank you, sir. Thank you, Dr. Hathi. Thank you, Dr. Mittal, Dr. Shrija,
Thank you
ma'am,
all.
everybody.
Thank you.
We'll join tomorrow at 10 o'clock in the morning.
Yes. Request everybody in the YouTube and GoTo, please join us back from tomorrow 10 a.m. onwards with the same link that has been provided to you. Thank you so much.
We'll also share feedback link. So once you fill the feedback, you'll also get e-certificate. So kindly join tomorrow in the morning. You'll also get chance to get e-certificate.
Thank you.
Thank you, everyone. Thank you.
Thank you.
Thank you.
Yeah.
बस ये